Thanks. Time as any, Mr. Newman. Take her down to periscope depth.
Yes, sir. Clear the bridge. Dive.
Stand right.
Awesome. Thanks so much. We're live. Yes, we're live tonight. Guys, we've got a really, I think, very important broadcast tonight. We're going to have a lecture with Salvador here. Beforehand, I want to thank him for giving us his time. We've also got John, Jason, and Praise here as well. We're going to be sitting back. If we have any questions, we'll do so. We'll have a short Q&A. After the fact, this lecture is going to be on junk DNA, ENCODE, DNA function. I know Salvador has more of a technical name for it. I'll let him cover that as well. But yeah, we're really excited for this lecture, Salvador. So thank you for giving us your time tonight.
Yeah, thank you. It was awesome. Really appreciate it. I'm really looking forward to it.
Well, if I could share my screen, and I'll just get to it. I won't even introduce myself. I'll just start talking. That's all right with you all?
Yeah,
of course.
It's fine.
At this point on our
knows you.
channel, you're no stranger to our viewers and listeners. So we really appreciate all the work that you're doing and everything that you teach us. So once again, thanks so much.
Can you see my screen? It says 4D nucleome, E4 epitranscriptome.
Yes, I can see it.
Great. I believe true science is a gift of God. When our Lord and Savior Jesus Christ said, "Consider the lilies of the valley, they neither toil nor spin, yet I tell you not even Solomon was arrayed as one of these." I think of the wonders of the cell. Evolutionary biologists like Dan Grauer tell us genomes are mostly junk, especially the human genome. But this presentation will suggest how genomes are amazing machines, thus affirming the sentiments that even the lilies of the field are arrayed in a way far greater than Solomon in all his splendor. So I've opened with a theological thought, but the majority of my formal presentation will be almost pure science in terms of observation and relevant experiments criticizing the claims of Dan Grauer and evolutionary biologists who are on his side. Maybe one way to introduce the theme of
Good boy.
this
Stay there.
presentation. This is a slight modified quote from the movie Wrath of Khan. And Mr. Spock says to Captain Kirk, he, that is Khan the enemy, he is intelligent but not experienced. His pattern indicates one-dimensional thinking. The original quote was two, but I think you get the idea. So my strategy for trying to show why Dan Grauer is wrong is his thinking is one-dimensional, whereas the modern thinking for the human genome is in 4 dimensions, at least 4 dimensions. If we add other dimensions, it's even more. So we have the 4D nucleome plus the E4 epitranscriptome projects. These are funded projects. These are not just buzzwords. They are funded projects. So this is a quote by Dan Grauer. He said, if ENCODE is right, evolution is wrong. So this kind of sets the stage for what I'll be talking about. I'll actually be trying to argue that ENCODE is right. So independent of evolution, we can kind of discuss whether ENCODE is right, just from the science standpoint. Now, ironically, Dr. Sanford said Dr. Grauer is totally right at his NIH lecture. Slightly different, but that's definitely the sentiment. Just kind of on a superficial level, if we just take the number of gigabases in the human genome, that equates to about 6 gigabits of Shannon information. That's based on the idea that each nucleotide position is roughly 2 bits. So that'd be 800 megabytes. If we take Dan Grauer's view that approximately only 10% of the human genome is functional, that would equate to 80 megabytes. So a rhetorical question, do you think something as complex as a human being can be coded with information as little as 80 megabytes? So that's just something to keep in mind. That's the intuitive thing. That's the first kind of punch I might throw at the idea that the genome's mostly junk. So what is the ENCODE project? It was a follow-on to the Human Genome Project, which completed around the 2000-2001 timeframe. It was headed by Francis Collins. So they found a good swath of, they say pretty much all of the genome was mapped. That is the nucleobases in the human genome. But they didn't know exactly what they did. So they started the ENCODE project, which had a smaller budget, which was actually not, maybe it was just a pioneering project. And it was actually started by Francis Collins. A lot of people resisted him in doing this, but he went forward. So that's also his brainchild. Even though his name's not associated with the ENCODE project today, he was one of the pioneers that motivated its development. So ENCODE has had a follow-on called the 4D Nucleome project. But that's actually that's only part of the picture, because ENCODE has given birth to other projects, such as these. The ENCODE budget, can you all see my mouse here, was a total investment so far of about $500 million. One of the parallel follow-on projects was the Roadmap Epigenomics project, and also known as REMIC. That was a $300 million follow-on. I don't know the budgets of these others, but they're throwing serious money at this because they're trying to cure heritable diseases. So this is from the NIH website on the 4D Nucleome project. And these are some things I'm going to cover tonight from the 4D Nucleome. So what did ENCODE actually do? What got all this started? Well, they wanted to know what the genome did, what parts of it did. They just basically had a parts list when they did the Human Genome Project, but they really didn't have a handle on what the individual parts did. So they just started doing these series of experiments, and you have in these blue boxes are the names of the kinds of experiments that they would conduct. And this went on. The people at the NIH would farm this out to laboratories, etc. And they were creating what is known as an encyclopedia of DNA elements. That was their goal. There is another project not at all related to ENCODE called the E4. It's called the Enabling Exploration of the Eukaryotic Epitranscriptome, and that is a mouthful, so they just call it E4. E4. So we've heard about epigenetics, which is kind of the extra layer of information that is put on the genome. They're finding there's an extra layer of information also put on the RNA transcriptome. So the genome's DNA, and we have modifications there. The transcriptome is made of RNA, which comes from the genome, but it has modifications, and we call those modifications the epitranscriptome. And here is a little diagram of some of the modifications of the RNA molecules. And there's a whole encyclopedia of these too. And there's even known what is RNA editing. So there are just levels to this. This is probably also just as equally interesting, but we don't have the chemical power yet to and the instrumentation to delve into it. But we suspect it's going to be a very interesting world. So this is where a lot of the DNA that we may regard as junk, it will create RNAs that have all sorts of interesting properties. So just bear that in mind. I won't be able to cover it much in this talk. In fact, I doubt I'm going to get through all my slides. And let me check the time. Let's see. Okay, you guys kind of just keep me posted on the time, and I'll just go as far as I can. This was from Dan Grauer's university website. He put this on his university website, and he shows a picture of his granddaughter sticking out her middle finger, and he just says on his university website, you know, that the students and faculty could see, he says, "My granddaughter gives ENCODE the finger." All right, so there's this anger there, and he called, he was calling the people of ENCODE all sorts of names on his Tumblr blog. He calls them crooks. He calls them ignoramuses. He calls one of their heads the scientific equivalent of Saddam Hussein, and probably the ultimate insult, he calls the NIH director, the founder of ENCODE and the Human Genome Project, Francis Collins, he calls him a creationist. So I just had this little video.
It's only a game. Why you have to be mad? So he, Grauer, and some of his evolutionary biology buddies put together this paper criticizing ENCODE. It was just terrible. The tone was vitriolic. It was unprofessional. It was insulting to many. There were probably over 200 researchers involved, from Harvard, Yale, MIT, just all these research institutions, including the NIH, and just calling them names. The title of the paper is On the Immortality of Television Sets, Function in the Human Genome, According to the Evolution-Free Gospel of ENCODE. This was very uncollegial on his part, and people had noticed. Another journal called him out on his behavior, and they called him the vigilante, and that was from the journal Science, which is one of the most prestigious scientific journals. And they're noticing this is not right. This is not behavior appropriate to a scientist. Someone can be wrong, but the way to call them out is not the way Grauer is going about it, calling them crooks and ignoramuses. That's just very unbecoming. Just argue the facts. So the way I frame the discussion tonight, this is evolutionary biologists versus medical researchers. This is not about evolutionary biologists versus creationists. This is evolutionary biologists versus medical researchers. This is a fight people like Dan Grauer had started. So what did he have to say? His 2016 archive paper said, if 80% of the genome is functional as trumpeted by ENCODE Project Consortium, then 45 to 82 deleterious mutations arise per generation. For the human population to maintain its current population size under these conditions, each of us should have on average 3 times 10 to the 19th to 5 times 10 to the 35th children. This is clearly bonkers. just an amusing anecdote, doesn't have anything to do with this. I was pointing this out on internet discussion forum, and I said, that's kind of bad that a human female has to have 10 to the 35th kids. And someone interjected, what's the problem? What's the problem with that? By the way, the guy who said that is a psychiatrist, and he's an anti-creationist psychiatrist. Sorry, that was an anecdote. Had nothing to do with anything, but just a little humor. So Dan Grauer concludes, if ENCODE is right, evolution is wrong. And as I mentioned earlier, John Sanford said Dan Grauer is totally right. So the question tonight, is the human 90% junk or not? I gave already one reason that I suspect that it's not. Here's another reason that it started to motivate people. A paper came out. That's actually cited Francis Collins here. Francis Collins' paper in the Proceedings of the National Academy of Sciences, 2009. 90% of disease-associated SNPs, that is single nucleotide polymorphisms, which are mutations, point mutations in the genome, 90% of disease-associated SNPs are located in non-coding regions of the genome. Now, correlation does not equal causation, but intuitively, this is raising alarm bells that maybe we shouldn't call this junk. When I was at the 2015 ENCODE Users Conference, where there are all these scientists learning how to use ENCODE data, and they were talking and they're conferencing with each other, I'd hear one scientist get up and say, yeah, I'm studying this disease or this particular cancer. And what I find is that it's correlated with mutations in the non-coding regions. They could specifically identify non-coding regions. So that was a story over and over and over again. So that's why there's interest in the non-coding regions. So one can imagine the surprise when the ENCODE researchers were starting to publish and say, hey, we suspect strongly, we're confident to declare that we think 80% of the genome is functional. These guys were just doing their job. And then an evolutionary biologist like Dan Grauer, who had no business meddling in medical research, just started calling them all these nasty names. So I said, this is a very interesting development, and it did bother me. It did bother me. I said, this is not good. Excuse me, I'm gonna move. So there is, when we get those outrageous numbers of 10 to the 31st children, it's related to this formula, the Poisson distribution in Kimura's 1966 paper. We're not going to get heavy into the math. In the Q&A, if someone really wants to get into it, we can talk about it. But that formula was indirectly quoted here in Dr. Sanford's NIH lecture. I call it the bonkers formula, the bonkers formula. It is also quoted in other papers, and you see it like in Adam Eyerwalker and Keith Lee's paper. By the way, what I encourage people, creationists, when they're trying to defend Dr. Sanford's work, I encourage them to say, this is not about what Dr. Sanford says. Dr. Sanford is only one among many geneticists. Every top geneticist on the planet that studies the human genome will actually agree that it's deteriorating. That's not isolated. To try to, these internet evolutionists will try to make it out that this is only Dr. Sanford's thesis. It's not. It's many others. It's only fair to point that out. People like Michael Lynch and Alexei Kondrashov. He even wrote a book, The Crumbling Genome. Brian Sykes at Oxford. Gerald Crabtree at the Howard Hughes Institute and also Stanford University. And last but not least, Nobel Prize winner Hermann Muller. Now, Muller was the one who came up with what I call Muller's limit in this journal in 1950. And his limit was the number of mutations. The limit is one mutation per individual per generation. There's some nuance between the haploid and the diploid mutation. That's not that important. But compare the limit that he postulated is only one mutation per individual per generation. Grauer accepts that he's willing to accept that there are 10 mutations per individual per generation that are functionally compromising. And if ENCODE's right, it's on the order of 80 to 100. There have been suspicions that it could even be in the thousands. But the point is, even the minimum number is 10 times beyond the Mueller limit. Now, I'm not going to mathematically demonstrate the Mueller limit, as there's a rich literature that's associated with it. I'm just going to argue from intuition. So just take an intuitive argument for what it's worth. It's inexact, it's simplistic, but it arrives at pretty much the same number that Mueller did. So I hope you guys have had something to eat. These are my some of my favorite foods: tiramisu on the left and a McDonald's bacon double quarter pounder on the right. The ingredients list for tiramisu is eggs, mascarpone, espresso, ladyfinger, sugar, spiced rum, and cocoa powder. Could we, through random mutation and quote-unquote natural selection, evolve it into some other list of ingredients for another recipe? And you all probably know the answer, but I'm just going to walk through this exercise. So, by the way, this is Herbert Spencer on the right. He uses the word, he's the one who introduced the phrase survival of the fittest, which Darwin himself adopted in his later versions of Origin of Species. So let's try to do this by a process of random mutation and natural, quote-unquote, natural selection. So I'm just going to do kind of like a very simplistic population here. So let's say that the original recipe is this, and we'll call it the parent in the first generation, and the parent has children like this, except the children have mutations like that indicated in red. So the problem is, what happens if natural selection takes place? Well, we're going to select only from the best of the worst, so to speak. It doesn't matter what we pick. All of them have some defect. I'm just going to pick child number 2 as the fittest. And child number 2 will have her children. And these children will inherit the parents' mutations, and each of the children will have their mutations. So we could see the mutation is not being filtered out by survival of the fittest, and the mutations will accumulate. There's hopefully no point in continuing. We know the result will be just a total mess. So the question is, could we evolve the ingredients of tiramisu to some other set of ingredients? I hope the answer is clearly no. There's some subtleties to this. If we have an average mutation rate of, say, 1 per individual, that would imply we could have some with no mutations, some with 1, and some with 2. The Poisson distribution covers that. So when we work through the numbers, we end up getting this table here so that we could at least, if we had a certain average mutation, diploid mutation rate, we could estimate how many females at minimum we would need, even assuming 100% efficiency of selection to keep the genome of that species from deteriorating. That was based on Kimura's bonkers formula that I pointed out earlier. So for even with just 1, each female would need to have 5.4 kids, and for 2 it'd be 14.7, so on and so forth. And Dan Grauer pointed out for 80 mutations, each female would need to have 10 to the 31st kids. So I'd like to just pause here if anyone would like to have any comments before I move on. So that's just setting the stage.
And any questions from anybody, I guess, on the panel? Yeah. I'm just fascinated by the talk so far. I mean, just how it sounds like so many people have underestimated the functionality of the genome. I mean, if less than 2% of the DNA actually codes for proteins, what's the rest doing? And kind of as you hinted at near the beginning, Sal, like a lot of these non-protein coding RNAs are now known to regulate virtually all levels of gene activity in the genome. I guess one question that I would have is regarding the criticizers of ENCODE, how they will criticize the definition of function that ENCODE used. Because as you were talking about how ENCODE, the results suggest that over 80% of the genome is actively transcribed into RNA, suggesting some type of function and activity. How does one respond to that type of criticism?
What I do, I mean, me personally, I don't get into definitional arguments over function. I really just try to avoid it. I said, this is all a red herring. The point is, do you think it's going to be a good thing or bad thing to have this change? And that's over time.
Right.
And what I've said, I don't even try to argue that the whole genome is functional or 80%. I said, what's so bad about saying we don't know? Is it such a terrible thing to say this is a working hypothesis? Because we have all these medical researchers, like I said, 90% of the heritable diseases are indicated in the non-coding regions. So let's just have a look. Why do they have to be called names? Why do they have to be called crooks and ignoramuses? This is so degrading. And these guys are trying to save our lives. So that's what I'm taking issue with. I said, this is a lot, this whole thing about function, this is maybe not a nice thing to say, but maybe I'll say it anyway. These arguments, when they start, this is like throwing up red herrings when you don't have really a case. Right. When the data are really saying something that should get your attention, and they're trying to suppress more investigation. I mean, what is the harm in trying to learn more of how the genome works? And so what if someone is mistaken? It's not like evolutionary biologists have been right all the time. So, I mean, so that's how I would argue it. I say I'm not even going to define function. Let's just keep looking. Let's try to keep understanding. Let's keep running experiments and finding, because we're trying to solve cures here. These are just sideshows. These are just total sideshows that are irrelevant. So sorry, I'm quite a bit more animated today than I was on Friday talking about the origin of life, because I think this is a gross injustice to the scientific enterprise and medical research. Yeah,
I think you put it perfectly. You hit the nail on the head when you say it's a red herring. That's exactly what it is. I mean, it can't be a bad thing that we're finding more function in the genome. And I guess one other question I would have before we move on to someone else who may have a question is, you spoke a lot about, Grauer there and his claims that, I guess with his most recent paper, I think it was in 2017, where he was saying that no more than 25% of the human genome can be functional. Is he saying that based on with the mathematics that he's looking at? Is he using like a deep time evolution starting point? Because he more or less is he talking about that in order for human populations levels to be sustainable, no more than 25% of the human genome can be functional because of the effects of harmful mutations. Like the more functional the genome, the quicker the genome would degenerate, I'm guessing, because the more functional it is, those neutral mutations would be more so nearly neutral. Like, can you expand on that a bit, Salvatore?
Unfortunately, I can't, because I stopped reading him. And this is also not a nice thing to say. It looks like the scientific community is not publishing his garbage anymore, really.
That's good.
He's relegated to publishing on these internet archives, not getting in peer-reviewed journals. And I don't blame editors for not wanting to touch his stuff, because it's not relevant. How does that help us cure cancer or heritable disease or diabetes? Really? I mean, if I went to a medical researcher, is he going to care? And I could tell you, I've hung around enough ENCODE researchers, not ENCODE, but NIH researchers and scientists. They don't care about what Grauer has to say. They never cite him in any of their research or their lectures. He's irrelevant. He's irrelevant. Right. And you know who's actually given him relevance? Unfortunately, it's the creationists. I mean, we need to stop we need to start ignoring him. And unfortunately, I had to give him some space here just to give a little bit more drama.
Yes.
But he's. I'm sorry, I'm going to offend a lot of evolutionary biologists, but their work is irrelevant. It is so irrelevant to these questions. I don't know why these guys are doing all these phylogenies on, this creature evolved from that, when it doesn't really have a lot of relevance to what the mission in medical research or biotechnology. It really doesn't.
So, quick question for you on the phylogenies and such. To me, when I look at that, the entire concept, it's almost like they're coming up with hypotheticals of, oh, well, if all of these changes happened, then it could be this. And is that a fair? I mean, I know there's more to it, but to me, it's at the core. That seems like what is going on there.
I wish I could answer your question. I've given up. I feel I've wasted too many years of my life studying evolutionary biology, and I've studied it also at the graduate level. And I really wish I could have answered your question.
No. The reason I bring that up is as you're going to be, I know you're going to be diving in much more detail on this topic here, and the details here in just a second. And to me, when I listen to, and this is more in the internet atheist debates and such, when I hear them making these arguments, I'm like, you are literally putting forth a hypothetical and then extrapolating based on your own hypothetical situation or scenario. And I'm like, how I can come up with anything that could hypothetically end up in this, especially when, we talk about things of, as you and I discussed, my background's in technology and software and such. And I'm like, yeah, well, I guess in theory, I could come up with a scenario where all of these different lines of code modified and ended up in a new function. That's irrelevant to whether or not it's a plausible supposition to be making.
And worse, it's not testable. There's no way to really test it empirically. I mean, it may be just the best phylogenetic tree you can come up with, and it sort of, quote unquote, makes a prediction. Like, I could make a prediction. If we evolved from bacteria, there has to be some creature in between a bacteria and a human being. So I'll just pick a eukaryote that has some similar genes. It's like, well, yeah, prediction fulfilled. It's not much of a prediction. I mean, so anyway, I'd like to move on maybe to the more interesting stuff. So thanks for the questions. Could we save it for later if there were any more?
Of course, yeah. And I want to say I really appreciate you answering those questions, Salvador. So yeah, continue on.
All right. So back to Mr. Spock. He said the opponent is intelligent, but he's thinking in one dimension. So we have to think of DNA in multiple dimensions. And 4D nucleome, as the phrase suggests, is looking at DNA in 3 dimensions and also in the time dimension. And one way to look at DNA, we tend to think of DNA, and it's in the textbooks, is you'll often see DNA is a blueprint for life. Well, really, it's a blueprint for the proteins of life. And I actually people are beginning to suspect that a lot of the heritable information is in the cytoplasm and even in the membranes, but that's just a story for another day. I mean, it really could be that DNA is just scratching the surface of the information capacity inside the cell. Because if you include the cytoplasm, it's going to be gigantic, especially the sugar molecules. And they're talking about sugar code. People can Google the sugar code. So we may have only hit the tip of the iceberg. But that being said, 4D nucleome, if we look at DNA as a scaffold, as a material, as a physical material, to park molecular machines, we begin to see how things could be functional. Because up until then, we've just been thinking of DNA as coding for proteins, maybe some regulation. But now when we start to think of it as a scaffold, it's different. Another metaphor, and as imperfect as metaphors are, these are the best I could come up with. We can also think of DNA as designated parking spaces with addresses. So you see these parking spaces have names to them. So the spelling of the DNA letters is important because that serves as an address. So think of it as addressed spaces. In DNA world, we call that names. We call these things motifs. So these are motifs that machines will search out for to park the machine. And I'm going to illustrate that. Most of the panel here probably is familiar with chromatin, but for the listeners here, I'm going to have to cover some basics about DNA. In organisms like humans, eukaryotes, we have an architecture like this called chromatin. So this is how our DNA is organized in eukaryotes. Bacteria do not have this form of organization. So here we have a chromosome, and if we start to pull it apart, it unwinds into these wires or these strands called chromatin. And chromatin is composed of DNA. Excuse me. It's composed of DNA that wraps around this bead-like structure called histones. So DNA wraps around things like histones. So it's like a bead. People will refer to it as beads on a string, where the string is DNA and the beads are the histones. And you can actually see here the DNA expanded out. Everyone okay with that? Now feel free to stop me as I go through these slides because they are technical. If I miss something, please ask me a question to clarify, because I sometimes will misstate things. So we have this, you'll notice this, it says here, this methyl group. That is a modification on the DNA. It's like a little post-it that's stuck on the DNA. That is what we call, that is one form of the epigenetic mark. It's an epigenetic modification. It's just like adding a little extra chemical there to the DNA. That's one form. Another form of epigenetic mark is changes. We can add little chemicals, like little post-its, to the tails of these histones. So you have these histones here, and you have these little tails protruding, and we can add chemical markings to these. These are epigenetic markings. Let me cover the methylation marking here a little more. And this is all related to the discussion about making if DNA is functional. So we have here a cytosine molecule, and it can be modified by a methyl group. There's some question whether some of the other molecules can be modified too, but so far the cytosine is like the dominant one in our literature. So we can add a methyl group. And so this is, if you imagine the red here, that's the DNA double helix, and you can have these little methylation marks. So this is reminiscent of random access memory. By the way, the name epigenetics, I believe it came from this gentleman here, Waddington. And here's another picture of some epigenetic changes. I will say this, I don't like the word epigenetics anymore. At the time that it was coined, they didn't have all the chemical details. So now that we have the chemical details, we don't have to call it epigenetics anymore. We can say, okay, this is a methyl modification on the cytosine, methyl modification on DNA. Then there's no confusion about what you're saying. But this is a little introduction to what epigenetics is all about. Unfortunately, there are a bazillion definitions of epigenetics, and they don't agree with each other. So I'll try to just be more specific. So I'd like to show what happens with these methylation changes. This is a box A here. They're able to make these very interesting graphs where they, I believe they use luminescent proteins that would, or antibodies that would bind to the methylation marks, and then you can illuminate them. So we have a zygote here, and it's at the stage where the dad's DNA, the paternal DNA, hasn't yet mixed with the maternal DNA, and you can see the methylation marks by the illumination there. And that's at 3 hours from after fertilization. At 6 hours, it looks like this. But come at 8 hours, notice the paternal DNA has erased. It's like done a reboot of its methylation marks. See that? Hope that's clear. And then sometime later, it continues. And then when the cell enters the zygote enters the metaphase, you could start to see I guess these are the chromosomes starting to break apart, and they're going to do their thing. Ignore the middle panel. That's just the control region. That kind of makes the diagram confusing, unfortunately. But then when we enter the 2-cell phase, you could see the changes. You could start to see the maternal DNA that is methylated, merging with the paternal DNA, and this is the 2-cell phase. We get to the 4-cell phase, and you could see the DNA, the methylation marks turning off. So what does that remind you of? That reminds me of random access memory. It's almost like a punch card. In fact, some of the diagrams that list the methylation marks between cells looks like this. I got this from, I know some people will say, well, where's your citation? Just Google it. If you search far enough, you're gonna keep getting all sorts of material. And I often quip, whatever I say right now is probably 5 minutes obsolete because the field is moving so fast. So you could see that the methylation markings are different between different kinds of cells. And from the Stem Cells Handbook in 2013, they used the word random access memory for epigenetic memory, random access memory. So I used to get flagged on that. They used to say, Sal, you're just making stuff up. And so I Googled real hard and had to look, and there it was. Other biologists were thinking the same thing. So great minds think alike. I kind of rubbed it in their face. So here is also some visualization of how this epigenetic memory, this random access memory. So this is really amazing. DNA is considered like a read-only memory, but there's a layer of random access memory on top of it. So we have 3-year-old twins here, and these are their methylation marks. And then we have 50-year-old twins, and you could see the change in their methylation marks. So I'd like to pause if anyone had any questions or comments. I hope those were interesting pictures. I don't know if you've seen them before.
No, I think it's, once again, it's so much fascinating. I find the function of the genome so fascinating, just as you were talking about chromatin, how these non-protein coding RNAs from introns actually influence gene expression by modifying chromatin, for the audience sake, as you explain that complex combination of DNA, RNAs, and proteins, right, that make up chromosomes. But for me, no specific questions, just once again, so much fascinating information. And John, Jason, Praise, any questions or comments from you guys?
Yeah, I'm just enjoying listening. I had a question, but I'd forgotten that as usual. But yeah, I know I'm really enjoying it. I'm really enjoying watching and learning about this. And there's a lot of new terms in there that I want to write down later and get timestamps and stuff. So I was really enjoying it, Dr. Cordova. You're doing a really good job.
Well, thank you. I'm only Mr. Cordova right now. But I do have 5 degrees though, so I have kind of a broad education, not specialized.
Oh, Dr. Cordova, sorry.
Mr.
Yeah.
Cordova is fine. I'm not offended.
No, I'm sorry.
No need to apologize.
If there's no more questions, we can let. I do have a question. Do you prefer to be called Salvador or Sal?
Just call me Sal.
Sal. Okay.
Okay.
It's nice to be called Sal. My detractors call me all sorts of names, so it's refreshing to be called.
Same here.
Well, you're doing a great job so far, Sal. You can continue whenever you're ready.
So this is chromatin. So I covered the methylation. That's one form of epigenetic change. The next form of epigenetic change will involve the histones. So these are the histones. These are beads on the string. I like this beads on a string. So just think about it. This is random access memory with beads on a string. I'd like to compare some of the historic man-made memory with this. So on the left side is what I call God-made random access memory. This is a picture of chromatin right here in an electron microscopy. So you see the beads here, like that. And you can barely see the wire. That's the DNA that's between them. So that's beads on a string. That's God-made memory. I'd like to contrast that with man-made memory. There was an old 1950s core memory that used magnetic core. It still sort of looks like beads on a string. And even farther back in ancient times, we had kind of beads on a string. There's sometimes they actually did use strings, not just wires or sticks, beads to have memory. That's an abacus. So that's kind of, I'm a very visual person. And so I just found this compelling personally. So I call that God-made random access memory. This is another depiction of chromatin. I'm sorry.
I said, yeah, wow. I said, that's amazing. You know, that the way that's set up like that, that just couldn't be anything other than made by a creator.
Isn't that cool? That just
Yeah,
amazing
it is. It's amazing.
Yeah,
It's
yeah. I think the Lord has a sense of humor. I just wow.
Yeah. Yeah, sorry.
Well, we'll go on. I'm glad you're enjoying it. So the reason I had this other picture, I wanted to highlight some of the more fine features of the histone. They're actually, the histones come in pairs. They're identical. And so there are 4 identical pairs to make 8 histones, but we usually kind of tend to think of them as only 4. But they're actually 8. And I'm just gonna take what's in the circle and I'm gonna kind of expand upon it. So these are the histones, but I'm focusing on the tails. I don't know if, let me go back. So you see these tails out here? This diagram is going to expand the tails a little more. And so like you have histone 3 here, and you could see all the different kinds of positions here on the tail, and they can have these little post-its, chemical modifications. The Me is for methylation, P is for phosphorylation, AC is for acetylation. I don't see any, there are not too many ubiquitinations, but they do, oh, here's one UB modification there. And just for completeness, I want you to see how the histone is spelled here. This histone 3, let me go back one. So you have this histone 3. I'm going to spell the protein out in terms of its amino acid letters. It's only less than about 140 letters. And the numbering of the histones is a little unfortunate. It's a little different. Everyone has their own coordinate system. But I'm going to number, this histone is, this K here, which corresponds to a lysine amino acid, is position 27. So I just go from 0 all the way up to 27. That corresponds to this one here. Do you see that? I have the orange mark here and the orange mark there. I'm going to show all the machinery that it takes to add a post-it note to modify that. So it's just like flicking a bit. So this is also another form of random access memory. We can be flipping bits, which is really cool. Does that mean chromatin is a computer? Some people have suspected that it is one of many computers in the cell. And indeed, they borrow language from computer science. So when we're flicking all of these, we're making these chemical modifications here, we say we are writing, we are erasing, or reading the mark. And they have all these chemicals that do it. So we have readers, writers, and erasers, very similar to the computer read-write heads in our machines, like say like a disk drive, except that the difference is in biology, you have multiple read-write heads that are just floating around inside the cell. And what's really amazing, it has to navigate to the right position. And I'm going to show that just for completeness for the chemistry nerds out there. This is how the methylations work. I just threw that in there, but we don't need to go there. We can kind of abstract out all the details. So again, we have a chromosome, and we have these, we call this complex. We have the histones with the DNA wrapped around. By the way, we also call this little unit a nucleosome, nucleosome. And by the way, that sounds very similar to nucleome. So when you hear 4D nucleome, these are similar words. So how does it navigate? And these readers, writers, and erasers have to navigate to the right nucleosome particle, the right histone, the right section of DNA. So I'm going to show that. So let's start with chromosome 12 here. Chromosome 12, let me highlight it. Chromosome 12 has a section of DNA that we call hot air. So just imagine we found this, we were looking at chromosome 12 and we found DNA that looks like this. And we have a name for it. It's called the hot air link long non-coding or long intergenic non-coding RNA. It's actually a DNA that codes for an RNA, so we call it hot air RNA. So how does this work? So remember, I'm just going to flip one bit. I'm going to flip one bit on a histone. So we start with the hot air link RNA. It comes out of the chromosome 12. It somehow navigates the chromosome 2 through all that colloquially what I call Brownian motion. That's not the correct formal term, but it is a it's close enough. It illustrates the point. So it's going to somehow navigate without any active propulsion in the nucleus in the cell. It's going to find its way to chromosome 2, and we actually don't know how it does that. That's so far it's like black magic. We have some idea, but right now it's pretty mysterious how it can do that. That's an amazing problem in biophysics. So that hot air RNA, it'll find a section on chromosome 2 that has a place just for it. It's like a named address. I'll just call it the hot air space. So that's what I'm trying to illustrate here. There's a parking space just for it called hot air that corresponds for the hot air RNA. When it arrives at chromosome 2, it'll trigger the assembly of this massive complex here called the polycomb repression complex 2. Sorry, I'm losing my voice. I'm going to have to take a little break here and let you guys talk. The black wire here is the DNA. That's DNA. And then these are the histones here. So it's going to find the right chromosome. It's going to find the right parking lot on the DNA. It's going to find the right section of DNA. And it's going to find the right histone. It's going to be histone 3. And it's going to find that methylation mark of position 27. It's going to have all that machinery. Isn't that cool?
Yeah, it is. Yeah, it's amazing. I mean, like, sorry, Stanley, go on.
Go on. And you were Jared? Did I get your name
Jason.
right? Jason. I'm sorry.
Yeah.
Jason.
Yeah, it's amazing how all this stuff, links together like this and works in conjunction with each other. It's doing stuff in the genome. The genome's working. I mean, the genome works on first, second, and third dimensions change over time to make the genome 4D, and just the unbelievable complexity and just in a so-called simple protein is mind blowing. It's and bits of proteins and just even piece. I mean, for these things to be like this is for anybody to say that just happened like that, like a friend of ours, Neff, always says, Nephilim Free. He always says they'll look at a pile of compost and they'll say a pile of compost is designed, but then they look at something like this and they say it's an accident.
And so as you're trying to move on here, you've mentioned the term machines throughout your entire discussion. Something I've run into on a very regular basis when I make the argument about the nanotechnologies and nanomachines that are enabling this read, write, edit process you're talking about. It's immediately dismissed as nothing but just random chemical reactions. Is that no, we're talking about real life nanomachines. Is that premise accepted inside of academia that yes, these are molecular machines that are doing direct functions rather than just being this random chemical reaction that's somehow resulting in these things like methylation?
If you're at the University of Houston and Dan Grauer's department, yes, it'll be a random reaction and of no consequence. But what's happening is we're actually beginning to correlate it to changes. So that little change that I was describing, that correlates to changing the quality of the skin. So depending on how this is flipped, you'll get soft skin like in an eyelid versus hard skin like you get in the soles of your feet. It changes. So these changes matter. And just because we don't know what it actually does right now, because we're seeing all these enormous number of computations being done, and we don't know what it's actually doing in terms of how it develops the organism. But it takes a lot of research. It takes hundreds of people to get a really good definitive answer just for that one. I'll tell you, this modification here is famous. It took at least a year and maybe a staff of 20 people to figure it out. And it was really hard work. And that's just one modification on one histone in the Hox cluster. We don't know, there's so many other regions of DNA where you have other histones, and we don't know what those do. So there's suspicion. So sometimes they're dismissed, but you know how they stop being dismissed? By research. When an experiment proves that this is what it does, the people are silenced. I wanted to say, and I'm just going to wax a little philosophical here because this relates to the book I'm writing. The way that we actually started to learn about this was studying fruit flies. Fruit flies ended up giving us an understanding for how the human body worked. So how the fruit fly changed it had a number of like bristles or something. The way we could change the number of bristles was changing the corresponding histone here, but it gave us a clue of how all of this machinery worked. So that's why we call it the polycomb repression complex. It affected the flies, the bristles on the fly's legs or something. And yet it has a correspondence in humans, but it's like, again, this is theological, philosophical. It's like God gave us all these little we call them actually model organisms. That's why it's like, why are these medical researchers trying to solve human cancer by studying fruit flies? It's like, well, we have all these organisms that are simpler that we can kind of break apart and blow apart and do all these cruel experiments that we would never think of doing to humans. They're just like gifts. So all these patterns of similarity are gifts for medical research so we could understand we're fearfully and wonderfully made. So that's my little mini sermon. So this is how some of these discoveries were made. So that's how it may be dismissed today. And I will say, even 12 years ago, when people were saying this was all junk, I had no answer on the internet. But now it's so easy to fight them because research has advanced in 12 years. So that's how I would deal with that argument when they just dismiss it. I'll say, well, I'm going to wait on the Lord. There'll be something coming up because it's to His glory to reveal this eventually. So this has been hidden for 2,000 years, or ever since time began. We didn't know all these miraculous things. Now in the present day, we're learning it. God stored it up for us in the 21st century.
Well, I find it so fascinating that I guess the DNA in general, but especially the non-coding regions of our DNA, as John was talking about, they do function much like a computer system, and these, I guess the deniers, I find that the ones that say, okay, the majority of our genome must be junk, no more of it can be functional than 25%, as you've stated, Sal, it hinders scientific advancement and it hinders medical advancement. And typically I'll ask those types of people that if all of this is truly junk, if over 75% of the genome is junk, why is the cell even bothering with it? Because obviously that type of activity would be wasteful of energy and resources, and natural selection should have eliminated all of this wasteful product so long ago. Do you find that there could even be an answer to such a question like that, Sal?
No, I don't. I try not to answer them anymore because they don't even have the same story, because some people say this is neutral evolution. Others will say selection. I said they can't even get their story straight. What's the point, And whereas what ended up happening to me, it was really part of my job to study this. I said, what have I been missing out on? When I saw this like 4 years ago, I wanted to say hallelujah in class, but I couldn't. And then I'd tell my friends, I said, wow, this is amazing. We never see this discussed on the internet, and so now this is why I'm very grateful for you guys. You've given me an opportunity to share this because sadly,
we're enjoying it.
Yeah.
We really appreciate it.
We're
I just,
really enjoying it.
it's
Sorry
amazing.
for standing.
No problem. I find it amazing because what we are dealing with is a highly compressed DNA code that's beyond the best computer code. But I guess those that reject that type of thought process, they are hindering themselves from such fascinating discoveries. Because just like you said, this has been hidden for 2,000 years, and just maybe 20 years ago, 30 years ago, a lot of the pro-evolution arguments had to do with the junk areas of our genome, pseudogenes, ERVs, the ALUs, for example. But now all this research, paper after paper after paper, suggesting that these aren't just leftover remnants of viral infections or genetic mistakes. I mean, these are functional DNA elements, oftentimes important to sustain healthy life processes in the cell. So we can bow down and say, praise God for this, but the ones that want to reject that idea, yeah, they're just hindering themselves and hindering scientific advancement mostly.
My favorite.
Yeah, no surgeon ever went to a book of evolutionary origins to brush up on surgical techniques before a difficult operation. You know what I mean? Like so it's ridiculous. It doesn't help anything. It's just a huge waste of money. I mean, billions and billions of dollars that could be put into researching actual cures and doing actual good in the world instead of wasting it on, rubbish, fairy tales.
Yes.
And yeah.
Exactly. So one of my favorite proverbs is, "It's the glory of God to conceal a matter. It's the glory of kings to search out a matter." God has concealed this for us to discover. It's just amazing. So we live in a very, very special time. I mean, Christians would have longed to have seen what we see. We are so blessed. I'm going to point out some things.
Of course, take your time, Sal.
So the importance of the DNA sequence is that it gives a name. It gives names. So the sequence is important. And this will relate eventually to repetitive elements, but I'm going to point that out. And it's very important for addressing that we have the right spelling. So the spelling matters. And you can kind of guess that how do the readers, writers, and erasers, how do they find the right location on the protein? It has to be spelled right, because that's how it's going to navigate there, because otherwise it's going to grab another protein. So there has to be some uniqueness in the addressing. And that's why there's certain sections of protein that really, really, really count. You cannot be modifying these willy-nilly, because if you start to change even the amino acids in between, like between number 28 and 38, that could mess up the ability of the reading, writing, and erasing machines to locate. See, I'm just pointing, that's looking farther down the line. So the sequence is important. Yeah, here are the readers, writers, and erasers. Now, I showed a really simple chromatin computation with I was just modifying this one, okay? I've seen the diagrams get ever more complex. So I saw one like this. I said, I wanted, evolutionists wanted to debate me on the emergence of eukaryotes. So I threw this diagram up. I said, okay, so archaea has kind of like a pseudo chromatin, but eukaryotes, it's really special. For eukaryotes to do DNA transcription and translation, they have to do stuff like this. That's not even doing, that's just to getting it off the ground, because if there's a double-stranded break in DNA, it has to go through all these histone modifications and coordinate them. Huge. And I said, do you really want to debate me on this? You could have a system that was just all DNA, and then now you have chromatin. You have to have all these machines to do all the what they call remodeling and all the cross signaling. That debate lasts about 1 minute. Okay. So that's why I like debating I probably would have a very hard time debating Erica, because my specialty is not on the high end like primates, but I like debating the low end of the so-called phylogenetic tree. I said, this is how I would prosecute that. I'll say, how did this evolve? How did chromatin, how did the chromatin system evolve? Because you need a minimum complexity to be able to even do DNA translation. It's nothing like it in prokaryotes. And that debate lasts about 30 seconds. Sorry, just had to point that out. That's why I love chromatin. So now, if we do a calculation of the number of, we can do a calculation based on the number of histone modifications, the number of histones per nucleosome, the number of nucleosomes in the entire genome. And when we tally it up, we have about 80 megabytes of random access memory implemented in the histones. I'm not even including the methylation marks on the DNA. So I calculated it to be 80 megabytes. Little did I know someone else made that same calculation. I calculated 82 megabytes. Someone else said 80 megabytes. So close enough, right? 80 megabytes, that's the name of the journal and the title of the article, in case anyone's curious. Barbara Bryant at Constellation Pharmaceuticals, when she published this, she argued that chromatin is actually, the entire system is Turing complete. That is, it can actually act like a computer. So every cell is a computer, but if you say, okay, every cell is a computer, and it has random access memory, and there are 100 trillion cells, if you work out all the total memory, that's 10 to the 21st bytes of epigenetic random access memory that be modified in a human. It can be used in the human brain also. So the human brain has huge amounts of memory. That's only a fraction. This is the epigenetic, the genomic and epigenetic memory. It's going to be dwarfed by the glycome, and I don't have even time to cover that. So we are fearfully and wonderfully made. And let me see. So I'm going here, and I'm gonna I'm gonna take a pause at some point, because even though I have tons of other slides, maybe I'll save that for another talk. But he is intelligent but not experienced. His pattern indicates one-dimensional thinking. So I'm showing you a little bit more of a little different dimension of thinking. So what about endogenous retroviruses? So I'm going to highlight that. So let's say the endogenous retrovirus is here. I think my slides are a tad out of order. Look at this. This is a machine here called CAP1. So it's interesting how the names of all these machines come together, but they're usually very short names. CAP1. CAP1 is a very important machine. It is a master regulator of the genome. And you could see here, we have the CAP1 has to indirectly attach to the endogenous retrovirus. That is why the ERVs are important. They are probably designed, yes, they do end up being viruses that can migrate outside, but I believe the original design is that it served as something to get this CAP1 machine to locate in the right place on the genome to do regulation. And we're seeing that. That was the subject of this article. So you can look that up. And oh yeah, this was, so let me see. So there are more articles that talk about how the endogenous retrovirus, through the process of being connected to machines, actually serves a regulatory purpose. That would have been easy to miss 12 years ago. So in the last 12 years, this is all exploding. Now we're knowing how this works. And I see if I can get to the point. Oh, okay. So I know what I'm doing. I'm going to summarize. We can wait and pause a little bit and talk a little more. But what I've showed with the chromatin, I haven't even gotten to the three-dimensional structure, but this is just kind of leading toward some things we can talk about in the discussion. The physical and data structure of the 3D genome, and I'm going to point something out. They're talking about chromatin. These exotic properties necessitate modularizing three-dimensional genome into tree data structures on top of, and in striking contrast to, the linear topology of DNA double helix. So they're saying there's another layer of information there that's in the 3D, more so than just kind of reading the DNA, from quote unquote left to right. And DNAs make RNAs. We're not going to cover this. DNAs can generate a neural network. The RNAs are an ideal material to make neural networks. So what we wonder what all these RNAs do, we're only beginning to understand. And I talked about the E4 epitranscriptome. So I framed this fight, between evolutionary biologists and medical researchers. I think, I know this is mean. Since they're so mean to me, I'll be mean to them for a change, right? I think this is more like Bambi versus Godzilla.
Go for it.
So I'm going to take a pause here. I have more slides and I can kind of go into more detail if you like. But if you had any questions or comments, or if we wanted to take some comments from our audience who's been so patient to hear me, I'd like to thank you all for listening so far.
And your voice is, you said you had a sore voice before. Is that, are you okay with your voice? Is your throat getting sore?
No, I'm just getting a little congestion, but if you all give me a break and make some comments, I should be fine.
All right, well, I'll pass it on to John and Standing because they know more about this than me. So,
so something when you're talking about the random access memory and all those things, just going as you're discussing that, I think about the and we hear this on a regular basis, and folks we debate in the comment section and everything about how DNA is a horrible data storage medium. And to me, the things obviously the amount of data storage we're talking about here is mind-blowing. And I remember reading a paper, I think about a year ago, when they had recently discovered that the amount of data storage in the brain was now expected to be 10x greater than the previous supposition. And I think it was up to like 2.5 petabytes. They were thinking about just in the brain storage. And I remember reading, then there was a little line at the bottom that said, and this may not actually be a realistic expectation. It may be much more than this. And when I was thinking about it, I went and Googled it, and I think it wasn't until 2011 or 2012 that there were even 2.5 petabytes of data on the entire planet. And so I thought about that and just was like, okay. And then now I think we're up, it's exploded dramatically with, the growth of, video-based social media. But now I think it was, when I did the math, I think it took 3 people's brains to be able to store all of the data on the planet. Yeah. And then when you think about that in the context of the energy, the low level of energy that's required in our brain to process and store that much information, when you compare that to our best hard drives, it becomes a mind bender when you think about what is actually being accomplished from a data processing and storage perspective that we take for granted, and yet we have people on the other side arguing that it's crap and is not a good medium. And I just find that very interesting. And to me, that's a question I have for you is, do you think, this is my position, that they are just living in a complete state of denial and refusing to consider how ludicrous the argument is becoming?
I don't know. I try not to psychologize people unless I can't really relate to them. And I was thinking, what do you guys got to gain? I mean, you're just going to take something, make it a target, and say it's garbage. You're just going to assume it, and you're gonna go all out of your way to claim it is. Just like Grauer, and I can name quite a number of others like Avais and Ayala. And I said, you have nothing to gain and everything to lose, because your reputation is going to get trashed if someone finds out it's incredibly well designed, and they're not going to let you forget it. And I'm not letting them forget it, because it's bad enough that they were saying it's just garbage, and that's from an academic standpoint, just let it go. But when they start trying to trash people's reputations, guys who are medical researchers and trying real hard, I really think that's low. So I can't.
Yeah, it is.
That's just really low. And so they. I mean, okay. since you're asking opinions for what opinions are worth, I can't prove it. I think these guys are just whining. They're like sore losers. You know, they wanted to win. They wanted to win the bad design argument so bad. You know, it's amazing. It's like the purpose of your life, the purpose of people's lives, like Dan Gar's, to prove that they're junk. You know, if he wants to prove he's junk, I'll let him do it. You know, but don't mess with science. But that's his purpose in his life. He wants to prove that he's a piece of junk. It's like, that's fine. Let him do that for himself.
Cosmic accident.
That is it is very interesting how it has almost become my goal in life is to prove that I have zero purpose and am really a completely broken down piece of garbage. I mean, it's when you really go all the way to the root of the arguments that are being put forth, that is what you are arguing at the ultimate core, and there is no purpose. And then I often wonder, this is what goes through my head many times when I hear these arguments of, then what is the point of even trying to figure this out if there is no purpose and all you're going to find out is how badly something works? Like, you don't tell me you actually think that. Why would you go and try and figure out how something that doesn't work very well works?
Exactly. See, that's Grau's papers. He says these papers. He's writing paper after paper, saying, "I'm junk. I'm junk." I'm just like, okay, you have, you know.
Yeah, maybe you're junk, not us. I find it just so amazing how many of these classic, you could call them, paradigms are being changed and completely overturned. Like, as far as I'm concerned, the entire junk DNA paradigm has been overturned. But even that talk of synonymous codons, for example, like, in short, right, we've got 20 amino acids, 4 different letters, A, T, C, G. Like, there's all these different languages in our cell, and we're told that there's all this, you know, redundancy in the genome, and there's this redundancy in DNA coding for protein, for example. But now we know that those redundant elements, those third codon positions, they don't just tolerate error in the cell. And I talk about this a lot. You know, they're using the different redundant elements to slow and speed the process. So this level of information compression that you're talking about, Sal, into every single letter boggles the mind. I find it amazing. And I've said it, I think, 2 or 3 times in your lecture today. The evolutionists, I like how you framed it, evolutionary, you know, biologists versus the medical community, because it's so true. And they're hindering themselves from so much, not only understanding on how truly complex the genome is, but just advancements in science, in medicine. It's a real shame is what it is.
Were there any comments or questions from our faithful listeners?
Let me see here. I think as I'm looking, I did screenshot a couple. If you guys had any other comments or questions real quick, I'll see if I can find some. Or if you wanted to go through a few more slides.
Yeah. Yes.
So you can also do that. I know you said you had quite a bit you wanted to go through.
Yeah, I think I want to cover the ERVs and transposons more.
Sure. Yeah, we would love. I mean, we love that topic. So far, we love every topic you're discussing. So
yeah, we love that topic. Yeah,
and really, I should cover
ERVs
the details of the actual 4D nucleome. I've only kind of been skirting it, teaching about chromatin, because you need to understand chromatin to understand the 4D nucleome. So maybe just in fairness for the readers, so I can at least
what that's it. That'd be interesting.
So up until this point, I've just been sharing what chromatin is, and even that is just mind-bogglingly, there's not really a word, wonderful. The 4D nucleome takes it to another level. So this is an illustration of chromatin, and we have like a gene here. This is a gene, and this is actually not to scale. But this explains a lot of things like introns and anything else has been called junk. It can be a location where we can get a molecular machine to do computation and work. So there's a lot of computation in the cell trying to figure out what it is and what state it is, whether there's been an injury to my neighbor cell and what I have to do. There's just all sorts of things that the cell has to do. I mean, if you think about trying to build a brain, all the computation that has to be done
to be able to start to construct something. So John, I believe, was saying just compared to the internet. So just think about how difficult it is to build out an internet, all the decisions that have to be made. So it's kind of, that's what I think a lot of the cellular computation is directed for. So anything that we have maybe labeled junk, introns, link RNAs, whatever, they can be used as addresses. In fact, this is really interesting. They can be multiple roles. But one of the things is some of their sequences can be used as addresses, and then the machines can park on them. And this reminds me of a robotic factory where you have all these machines and they have the tools, and they can, it's very much like what I saw in that diagram with the genome. So right there is kind of already kind of, it's like, this is how DNA can be used. It can be functional. If it's for structure, it's not just blueprints for proteins. But what's really amazing is this is where we start to get to the 4D nucleome. We have different genes. They can be on the same chromosome, and they can co-regulate each other. So segments of a chromosome can be brought into close proximity together. They can be brought in close proximity together. They can share chemicals, and then they can exchange. They can kind of like quote unquote signal. It's not exactly the right word, but they can kind of communicate in a way, and they can control. They can co-regulate each other. So this is very beautiful here. It's also kind of amazing how they can actually be, there's not a lot of energy to be doing this, but they're moving and reshaping the chromosome so that the genes can form these little transcription factories. But what really blew my mind is that can be done between chromosomes. That was a big discovery, and this was not too long ago. I don't know when we talked about the hot air RNA. There's this FIRRE, the fire RNA, and it will bring together a segment of chromosome 2, a segment from chromosome 15, a segment from chromosome 17, and then a segment from chromosome X. And they're going to get together and do their thing. So this is you're starting to see how all these pieces can come together. One thing I should mention, what does this mean? It means that maybe the intron of one gene is going to help regulate the expression of, this could be, we don't know if these could be introns that are having molecular machines park, and it's going to regulate a gene on another chromosome. That's what we're finding. And this obviously takes a lot of work to find. So sometimes our detractors will say, well, we have so little experimental proof. And it's like, well, we're not going to find out more if we stop the experiments, because it took a staff of 20 people a year working just to get that one link RNA kind of figured out. There are probably 100,000 of these. Okay, so there's not going to be any shortage of work. And by the way, that was only in one cell type. There are hundreds of cell types in the human body, maybe more depending on how we define it. So that's a different way of looking at it. And now I'm going to show something. So we have all these different cell types. We have like a stem cell, which can differentiate itself into all sorts of other kinds of cells. So we have like cells that are neurons, nerve cells. We have skin cells, I think epithelial. Correct me if I'm wrong, someone. We have erythrocytes or blood cells. And then these other kinds, we have cardiac cells, blastocyst. I'm not familiar with this one at all. Chondrocyte. Okay, apologies for not knowing that. So how does it change? How does it change to, how does it know I'm going to be, a nerve cell versus a cardiac cell? How do I know that I'm supposed to be a neuron, a nerve cell versus a fat cell? How does it process itself? Because you could tell they look very different. Well, part of that involves the chromatin.
So the way these the chromatin in a pluripotent cell, so this is let's say this is a pluripotent stem cell, the way that it forms these kind of loose loops here, and it allows kind of a little bit more exchange of chemicals or things going on. But when it gets differentiated to be when that cell becomes either like say a neuron cell or a fat cell, the loops close, and that changes gene regulation. So gene regulation is done by geometry. I believe that sometimes the closing off will, it does something, and that's something worth studying. Like I said, whatever I say today is like 5 minutes obsolete already because the field's moving so fast, I can't keep track of it. But in addition, I should point out what's going on. In addition to all these loop changes, and that's what the 4D Nucleome project studies, they study how these loops change between cell types. In addition to that, you have all in parallel happening in the cell are all these histone modifications all along the chromosome, and there are a bazillion of these histones in the chromosome. That's changing, and then you have the methylation marks on the DNA. So all this parallel processing computations going on to be able to make a stem cell become all these parts, and that's how we become from that one zygote into an adult human that has all the systems that we have: the digestive system, the neural system, just the immune system. It just goes on and on and on. Just amazing that it could be all packed into one zygote. And here's another one that deals, it's describing how the changes in the looping structure work for these kinds of cells. I think that those look like kind of related to immune cells. An immunologist is just going to pounce on me for not knowing this. So we talked about the transcription factories. So I'm going to have to talk a little bit about the zinc finger protein to introduce how it relates to these loops. So to understand how all these marvelous loops come together, I'm going to just have to talk about a specific protein. It's called the zinc finger protein. So we have DNA, and we have a protein that grabs it. That's why it's called the zinc finger, and is first characterized in a frog. I think that's a frog. All these Latin names confuse me. Anyway, so we had this protein, and we noticed that it would connect to DNA. We call that DNA a DNA binding protein. So they called it a zinc finger protein. And I'm actually going to show a zinc finger protein. This is what it looks like. This is the spelling of the human zinc finger 136. And I think it's only spelled with like 500 or fewer letters, amino acids like that. And the fold looks something like this of his individual zinc finger. I'll cover it a little bit more so you could see it. So you can imagine, I'm just going to just kind of traverse these individual lines graphically, and you can see how it corresponds to this is the actual cysteine residue. The C is for cysteine, H for histidine. So if you just take these letters here, and it would spell like that. Do you sort of see that? Everyone see how I did that? I'm just going to take this line and spell this. So this is a two-dimensional representation of the zinc finger. The actual fold looks like this. This is what the structural biologists do. They obsess on how this is going to actually work. And there's a zinc ion here that sits there. This gives it this is magic, because it makes the fold very simple and very efficient. So each of these lines is a different zinc finger, and you'll see it when I get to some of the next slides. What I'm trying to convey here. So what's nice is you could actually see a little bit of the protein structure. This is a non-random structure in the protein, very obvious visually. Dr. Sanford taught me how to do that. I call it skittleizing. So we can name each row an individual zinc finger, and I'll show a picture. So I just put the letters here on the side, and you could see that each of these is a separate zinc finger. And the way the protein will be is, so you saw that earlier picture where you had a hand grabbing DNA. So we have these zinc fingers, and the fold will look conceptually something like this. The protein will actually lay kind of sideways like that. Okay, so every protein has its own fold. This part of the protein will actually lay like that, and it's going to connect to the DNA. Now, this is what's really amazing. This is why it's really important that the spelling of the letters is correct, because this is just like the address. So what the zinc finger, when this is programmed, it's going to find like a few letters here. And the next zinc finger will find some more letters. It connects. So this acts like an address. So you could see that the spelling of the protein is really, really important, because otherwise it's going to grab the wrong piece of DNA. And I've confronted evolutionary biologists. I said, how are you going to find in 3 gigabase pairs the right section of DNA? How can you expect a random zinc finger? It's just going to grab any random piece of DNA, if that, because it may have an incoherent array. This is like taking a random, just cutting out a key. And that's why actually I was showing keys earlier. Do I have pictures of keys? Yeah, I was saying this is like trying to cut a random key and expecting it to open a lock. It's just ridiculous. So this is the reason I like this is beginning to show the importance of why the spelling of the protein is very important, because we're actually now, men are trying to actually now start to make these artificial proteins, these designer-made zinc finger proteins, so they could do therapeutics. And they're finding how difficult it is to even spell just one line. That's not a very long line. They're having an incredibly hard time getting it to spell just right, so they could grab the right piece of DNA. And it's really hard to get it to all coordinate, because these zinc finger domains interfere with each other. It's not quite modular. So
quick
please.
question for you.
Yes.
So are you, if I'm understanding you correctly, not only does the sequence for the protein have to be correct for it to fold and exist in the first place, it also has to be correct in order for the function of that protein to work in relation to it being able to bind with the appropriate location in the DNA, which may be completely separate from the sequence it came from.
Exactly.
So it has, there's multiple elements of control that is being programmed not in the sequence from both how it is synthesized as well as how it functions. Is that correct? Okay.
Yes. So not only do you have to have, I mean, if we were designing this, okay, just hypothetically, if we had even the fraction of the skills that the Lord has, we would have to design the DNA string, and then we would have to design the zinc finger that's going to bind to it. Now, that may not be so bad if we're just doing this randomly, but to put this in the context of a working cell, amazing. This is the problem that has to be solved to get this to coordinate. And we can barely, some of our zinc finger proteins are only, the man-made ones are only 3 zinc fingers.
So where my mind is going with this is, this almost seems like an entirely different level of chicken and egg problem.
Yes.
You've just added, this is like the 4th or 5th chicken and egg problem for life, right? In terms of how it's come to be.
I'm going to just move through some other. Let me please keep talking. I'm going to show this in relation to an earlier slide. I'm not ignoring you. I'm trying to.
No, as you're looking for the slide, the something I've debate I had myself and Neff had recently. One of the things that I was putting forth was, what we're seeing, and I didn't even know about this second. I knew there were multiple subsets of data inside of the code, but I was making the argument that it's like if we were writing a JavaScript function, but it could be multiple functions simultaneously. And how, yes, that would be really cool to do. However, that would be almost impossible for a human to be able to figure out how to do that. Like, have that number of dual processes going on in their own head as they're trying to create the code in the first place.
Yep. And I, by the way, why this is fun everything I've said, this is the tip of the iceberg. I have more slides, but these, remember back to
Oh,
the
we'd like to see you come back for hours if you could. We could sit here for 5 hours, 10 hours listening to you talk. If you could talk for 10 hours, I mean, there's unlimited time here. If anything you ever want to, like any books you ever want to push, or anything you want to, like anybody you want to help out by mentioning, anything like that. So yeah, we
well,
we're happy to. But I know you can't do that because you've got a sore throat. But yeah, we could have you back anytime, as many times as you want. Have you back every day.
Thank you, Jason. It is well. Thank
Yes.
you. Can I tell you just a little as an aside? I had a conversation with Dr. Sanford like last year. He said, "Sal, you're pretty much invisible. No one knows about all the work you've done in the last 6 years." He said, "We need to pray to let people know more about what you know."
Yeah.
All the stuff I've written.
Yeah.
These are slides I've prepared, and you're like the first big audience I've shared. I've shared it to faculty at Christian universities, just to the faculty, not their students. So
that's a shame because they wouldn't understand it. Like, I mean, you're way above our heads. There's no doubt about that. And yeah, but it's good. And I hope that, and we don't mind if you have a whinge too. I mean, we're not the sort of channel, we don't like to get negative. If you ever feel like it, I'm not saying that you would, but if you ever feel like it, you're quite welcome, like, as you know, because you've seen us in the comments section. Yeah. And yeah, we're really enjoying it. I mean, I'm the sort of person, I'll go back later and I'll watch this thing and I'll take notes, and I'll do a write up on it and send it to Standing for Truth and Matt and give timestamps and stuff like that, and they can then they'll do something with it, and we could put your link, or if you want to give Standing for Truth some links from people or whatever, or books or anything like that he could put in the description, then that would be if you wanted to do that as well.
yes, maybe in the future. So I'm glad I could be of service just for today, and we'll just see where the Lord leads it. So this is,
and you can use this channel to advertise your whatever you want.
oh, thank you. I will take you up on that, and hopefully we as
We'd be happy to do it.
Part of the body of Christ, we can move forward as kingdom together.
So helping us as well. I mean, you being here is a great help to this channel, and it's a blessing from God to have you here. So we're happy for, thank you very much from the bottom of our hearts.
Thank you, and I'm grateful.
But if you want to go on, you can go on as long as you like, or you can just end it anytime you want, Sal. If you just decide that my voice is getting whatever, you can go on for another 2 hours. We're happy for as long as you want or as short as you want. It's all up to you.
I think I at least want to do another half hour at least.
Oh, great! That'd be awesome. I'll just mute now and let you go.
I want to hammer the people that've been criticizing the ERV argument, the endogenous retrovirus. I just really see.
Oh, I love
this
that.
is great. That I want to point out one other thing. You see, in school, for people to get to this point, because schools are, when they're training people to learn stuff like this, these are for biologists, professional biologists, chemists, pharmaceutical researchers, medical researchers. So they have to go through 2 semesters of general chemistry, 2 semesters of organic, 2 semesters of biochemistry. Then they get to this. What's been encouraging is in the space of maybe 2 hours, for people that aren't, I presume, don't have a lot of chemistry background, they can understand this. That's because academia is, to teach this, they're oriented toward, the medical doctors and the medical researchers, and that's great. It's not yet, no one's decided to build teaching materials to teach them about God's works. No. And this can be taught to someone who's like a freshman and without a lot of science background. I feel like you all are kind of getting what I'm trying to say, and without having to go into all the deep biochemistry and organic chemistry. So anyway, the endogenous retroviruses, we need a crab zinc finger to be able to have this complex here. The crab zinc finger, crab is a little extra thing for the zinc finger. It's a specific family of zinc finger. It has to be matched to the ERV. It can't just be randomly there. And it occurred to me, one reason we have ERVs splattered all over the genome, if you want to regulate multiple genes simultaneously, this is a really good way to do it, is to put ERVs there. And it's like, this is staring us in the face why it's spread all over the genome, and also with slightly different spellings. That enables, if we had master keys and all, it enables kind of hierarchical control. You can group the different gene sets differently with slightly different ERVs. Like if you have a master key, it can open several doors, and you can have families of master keys. This is actually the different spellings of the ERVs. They are like different locks. And you can have hierarchical control where you could have maybe one master controller that can go and modify all the ERVs at once, or if you just want them to do sets. That's not in the literature, but I'm feeling it. I'm feeling it. That's where the way it's going to go. So who knows? Maybe it's been rewarding that I can sometimes say, "Hey, this is a great idea. Let me see if someone's thought of it already." And it's like, "Oh yeah, they did." So this is ERVs, and I'll tell you a funny story about this. Evolutionary biologists were just saying, "Oh, these ERVs are random," and then they began to find that zinc fingers would be programmed to connect with them. And they said, this is really kind of interesting, because when a new ERV pops up, there's a new zinc finger to be right there. So they said, the solution is coevolution. And just like, okay, so that's how they're gonna do it. And I've tried to argue how ridiculous that is because of the difficulty. That's why I showed this zinc finger array here. I'm sorry that it's so far ahead. You could see the difficulty of reprogramming a zinc finger protein. This is a crab zinc finger protein right here. You could see the difficulty of having to reprogram this. If you have a different ERV, you're going to have to reprogram this. And if you're reprogramming this, a mutation in this region or this one is going to make that zinc finger jump to a totally different geographical region. Could be a different chromosome, totally far away. It's not going to, it's not.
Wow, I didn't know that. It can actually jump like that.
Yeah,
it can actually go to a different chromosome like that.
Yeah, it'd be a total.
That's amazing.
So how do they solve it? They just say, oh, coevolution. They never actually work through the details like I've watched.
Co-opted.
So basically, from a computer programming comparison perspective, it's like saying that both the call function and the data that's being called both came into existence at the same time.
Or one evolved, one changed first and then the other adapted. And I'm just like, well, that's all well and fine, unless the organism dies in the process. Or comparably, just think about this. Let's say you already have an existing function and you start modifying this and it doesn't bind anymore. Well, that'll cause sickness. We have literature that says, oh, modifications in this zinc finger has affected the connection. We call it binding. It's affected the binding of the zinc finger to the DNA. It's implicated with this disease. I'm just like, well, oh yeah, duh. That's, you sort of expect that. And so what if you had a gene duplication and then that zinc finger that has a different spelling and it starts interrupting either the processing of another zinc finger? I said, this is not good. If you start having a gene duplication and then you're having all these random mutations here and this machine is jumping all around the genome, this is not going to be good. And I'm just like, it's going to be selected against. What I try to say is natural selection prevents change. It doesn't, it can facilitate it a little bit, but it does a lot to prevent change because random mutations, bad stuff. I say bad juju, So I'm going to show, this is also related to, this is where it starts to get mind-blowing for the 4D nucleome. This is another zinc finger protein. It's called CTCF. This is how it's spelled. And you can see it's not quite as nicely arranged. You have to sort of force fit the letters. but these are zinc fingers, and we've been able to test it. These are zinc fingers in the CTCF protein, and I'm going to just highlight some other parts of the CTCF protein. There's a region here where other proteins can connect. It's called the Shark BSH3 connection. So when we see proteins connect together, and I had some pictures like that with the polychrome repression complex, the protein has to have, just like when you have parts in a car, it has, every part has that may connect to multiple other parts. It has to be shaped right. So here is this green region will connect to another protein, and then the zinc finger will connect to the DNA. So if you can kind of, I'm sorry I didn't, if I posted that picture of, maybe someday I'll get some pictures. If I go through this slide presentation again, I'll know to post pictures. So you can just imagine that this region connects to the DNA, this region connects to another protein. So this is just like car parts. So this is why it's really hard when evolutionists talk about, how random mutation can develop a protein. I just laugh. I said, well, it may be able to develop some random enzyme processing catalytic protein, but if it has to connect to other things, just forget it in a rational way. Because look at all these connections. It has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 specific connections, 11, and they have to be spelled right. So I'm just like, goodbye. This is why I like this. With structural biology, you begin to see why proteins have to be spelled right.
So this this puts another layer on the supposed synonymous codon argument of no, it has to be the specific one to result in the correct ultimate binding to whatever part it is ultimately going to connect with.
Exactly. So let's say, not that this protein, I know this protein doesn't connect generally to an exon, to a coding region, but if you had a zinc finger or any other binding protein that had to connect to a specific spelling of the DNA, the synonymous change will affect that. It's very well known in regard to microRNAs, not so much with zinc finger proteins, but we have literature on the effect of synonymous changes on the gene regulation of microRNAs. And I can't believe it, evolutionary biologists are still saying the Ka-Ks ratios, show that these are neutral substitutions, or the assumption, show that it does this or that based on the assumption of neutral substitution. And just that's nonsense.
So in the context of a microRNA, potentially could the synonymous substitution alter the silencing or the different variations of what are happening with microRNAs? Would that alter what the intended modification that's being executed by the microRNA? Yes.
Oh, and you did speak about that precise binding. I have a picture here. Let me see. It may be way down. I'm sorry, I'm jumping around. But
No.
Be the best way that I can answer your question. Oh no!
It always happens.
Let me bring. What did I do wrong? How do I bring
That's
that?
all right. Take your time.
Let me.
Our
see.
audience is used to technical difficulties, believe me. So it's not a big surprise for them.
Do you see my? Did it look like? Oh, here it is.
Okay.
Do you see it says here in exons as enhancers? Does that come up?
Yep.
All right. So you asked about binding. It just occurred to me, your question, John. This is why synonymous mutations can be bad. If you have an exon helps code for part of that's used to code for part of a protein, right? If it turns out molecular machines, this is the dual use. It can be used as coding, or it could be used as scaffolding for a molecular machine when that gene is turned off. So let's say that gene is turned off as for gene expression. It can be used It can be used in another context to be a place you can put molecular machines to do gene regulation on other genes. So you remember that? See, remember this picture here where you can even regulate genes on other chromosomes? So just imagine you have molecular machines docking on an exon that's not presently in use. Isn't that cool?
It puts this is on a much lower level than what you're discussing now, but I remember the first time I came across the premise that different transcription factors jump from one chromosome to another. To me that was mind bending, and that what you're showing us here is taking that to an entirely different level of, hey, cool, we can this same thing can have multiple uses, even if it's not being expressed in the way that we generally think about it from the layman perspective. It's still having a function, even if it's not turned on in the way that the common person thinks about.
Exactly. That blew my mind. And this is where this synonymous change will affect the what they call the binding affinity, if not the entire what they call binding specificity. So that molecular machine may not be able to park there, or park there with enough strength, or any number of things that could go wrong. So I wanted to show you that. So that so that's to answer your question. It will affect the this idea that synonymous mutations are neutral. This is one example that puts problems in that hypothesis.
And I'm assuming this also, the things that are being discovered now also put even the concept of point mutation into an entirely different perspective.
Yes, yes to all of the above. So going back to where I was with the CTCF protein, and if you have more questions, let me if you'll.
No, we're shutting up. Go for it.
It'll bind to this. The central part of the motif, the DNA motif, will be like this. It could be a little different on the edges, and that's a whole nother story that we're only beginning to understand. So this is why you need 10 zinc fingers. It'll need, connect the interior zinc fingers here will connect to this, and then you can have different spellings of these regions. And I don't exactly know how it totally works. I don't think anyone does. Again, I'll point out, you see this green region, and I should have highlighted it here. Where is that? What row? That was 1, 2, EPQ. So this region here connects to another protein. So you can kind of guess at the fold of how this thing is, get an approximation. So you have the zinc fingers here. You have another protein out here that called SH3 that's going to connect here, and who knows what other things connect around. But how this is relevant. So we had these loops here, and the loops were described in Christopher Roop and Dr. Sanford's book, Contested Bones. And it does mention this loop regulation. I contributed to that part in the loop regulation in the book, Contested Bones. So we talked about these loops earlier in cellular differentiation. I don't know that the gene is actually here. That could be, I'm still learning. But anyway, we do know that we have these loops. And we have this CTCF. See, that's the protein, the CTCF protein, which was here. Remember this protein, CTCF, and I'm just going to represent it in red. And this is mind-blowing here. So you have the CTCF protein, and notice it points this way, and the CTCF points that way. So if we are reading along the DNA in some arbitrary direction, you see the green arrows like that, like that. So you could see one CTCF has to point this way, and the other CTCF has to point that way. Meaning we could say that the CTCF has, the protein itself has a defined orientation. And the way it does that is the sequence has to be put in one direction. It's spelled here in the DNA, So the DNA has to have this little motif here put in there in the right way. And then it has to have like the reverse complement put here. So the CTCF you'll have a CTCF pair, one that points this way and one that points that way. Otherwise, the loop won't form. So there has to be foresight between this section and then this section. It can't be random. It has to be spelled the right way. So when they say these,
and it has to be spelled correctly in both directions, right?
correct. So when they say this is a random insertion, I'm going to give you the best part. Okay, I'm spelling it out here. These motifs are inside. Oh, by the way, this involves other proteins like cohesin and topoisomerase 2. B, that's the protein I study with Joe DeWeese, and I published on it. Sorry, just had to do a little self-promotion there.
That's fine. No, don't be sorry. Do it. Don't look. Anytime you ever want to do anything like that, just go for it. I mean, if you want to put a link to your channel, a link to your work, if you've got any work up on the internet, anything, just give it to us and we'll do it. We're not, motivated by, like it has to be, we're not like some other channels where whatever. We're happy to help you out like that. So promote anything you like.
Oh, that's so generous of you. I hope we can work together. That means a lot to me.
Yeah, I hope so. But yes, it's good. Yeah, man, and I'd love to get John and Matt on next time. Matt's just a bit busy today because he had a problem with his car and he was just So, but apart from that, no, yeah, there would be.
So
yes,
I can tell you this much: you will greatly enjoy conversation with Matt. He is. Epigenetics is one of his favorite things to research, and he is. We joke that he is our the encyclopedia of the team, and you and he could have you and he could have very high level conversations about this from a technical perspective, like very high level. You would not have to. I'll put it this way: you would not have to dumb it down for him at all.
Wow, that's great. It's great to meet you guys. So as headed, this is the killer. A lot of these motifs are in sign elements or ERVs.
wow.
So are you stating that a core component requirement for the folding that you're showing us now requires ERVs to be executed?
I wouldn't go that far. I'm saying occasionally we'll have this signature inside. It'd be sufficient inside. It's also it's really in sign elements, but it's in a few ERVs, and I have the citations. Oh, I put it somewhere. It was in the slideshow. I mean, I know I just blasted through it, but one of those did say CTCF. ERVs had CTCF in it. CTCF binding sites. So there are a few ERVs that have it, but they're in the sign elements. They're in these transposons. I haven't even covered the lines. Say that for another time, maybe when Matt's around. But this is what's killer. So to have all these, when you're having all these loops and they're saying, oh, these transposons are just randomly put out there. There's so many of them. Why do you have so many copies? Why do you have millions of copies? Well, they are nice targets for the CTCF protein. And the CTCF protein coordinates with cohesin and topoisomerase, and they make these loops. And these loops are important for regulation and cell differentiation. So I said, we wouldn't have had this. We were totally defenseless for the last 20 years. These evolutionists were just saying ERVs are junk DNA, they're parasites. See how quickly it's turned around? Someone's fighting for us. Someone up here is fighting for the creationists.
Quick question for you on this. So very often we hear about the fragmented ERVs. Please tell me that some of these binding sites have been found in the ERV fragments.
I can't tell you that. I don't know. I really don't know.
No, I was if that was the well, I'm guessing we probably haven't discovered a whole bunch of things so far, but in a week or so ago when Standing was debating conspiracy cats. He went on this whole cats went on this whole tangent about the fragments of ERVs all throughout the genome, and how that was a fundamental example of evolution and the time factor and such. And anyway, Random sidebar there. If that had been discovered, that would have been a hilarious thing to know. Of like, oh yeah, and by the way, so the reason there's ones that are fragmented, they're not actually fragments; they're actually binding sites. That'd be an awesome discovery if that happened to be the case.
I will say this: I've seen in my gene browsing, I have seen damage to the DNA. I mean, I have seen evidence of real pseudogenes. So we are intelligently designed, but also cursed, and so I'm not averse to thinking that we have shared damage, that we are created, but then when again, this is theological, this is not science. When man fell, they probably may have had shared designs that broke the organisms the same way that they broke us. So that's just a speculation. And
you mean like the animals broke the same time that we broke, or the organelles? Did you say?
Yeah, maybe the same ways too. In some. So
yeah, I think so too. Because, sorry.
Oh, go on.
Oh no, I just heard. I don't know if it's true or not. You probably don't know about this because it's probably not your specialty. But I heard, and I'd have to check this out. I don't have a paper for it or anything. That there's certain animals like that have faster generation times than us that are sort of breaking down at about the same speed as we are. I've heard that, but I don't have any actual hard evidence for that. I mean, maybe Matt does, or there's something in the books, but I don't know if you've ever heard anything about that.
No.
Oh, okay. So you don't get into that. You just, sorry.
This is just a passing thought, but I guess the general point is, I think there's been damage to the genome, and it wouldn't bother me if we got broken the same way certain primates did. It used to bother me, but now that when I study the difficulty of evolving one creature to another, I'm just like, if I had to pick between one or the other, I'd still pick special creation, because
especially
the more that I
get a
study,
rat.
it's like, do you think this could really evolve? It's too hard to believe. Too hard to be an atheist.
I have zero issue with any similarities that are found. To be honest, the
same here.
When you look at the point you just made, Sal, of this isn't going to happen without direction. Period. At the level, it's just beyond reasonable conclusion. But to me, the more complexity we discover and the more intricate, dependent parts and all these different things that we're discussing now, to me, that just lends even greater evidence in favor of a creator than evolution, to me. Of like, well, of course there's similarities. Why wouldn't there be?
Of course. Yeah, like monkeys have a similar body plan to us, and a similar, not the same, but similar. And if you see certain coding regions on an end of a chromosome that they say it was 540 zeros, one with 540 zeros after a chance of evolving being like that without evolution or whatever. Well, I mean, of course, because we got similar body plans and we look similar sort of creatures in a way, like in the way we look of arms and legs, and so if we have things that are in common like that, then it just screams a common creator, not a common ancestor. But I was looking at, there's 60 million gene sets that have been sequenced from organisms in the ocean, and they've only found 12 or 14 that may have a deep branching pattern and stuff like that. I mean, 60 million gene sets, and they only found 14. I mean, these sort of things are just, you have to be really deluded or focused on proving God doesn't exist to think that, those sort of things, like the evolutionists even saying now the bush of life and not the tree of life. And then that was coined by, I can't remember his name, but I saw the debate was in the Great Debate with Richard Dawkins. And he said that about the 60 million gene sets. I can't remember his name. I wish I could. Terrible with names, but yeah, it's mind blowing that people if I had known this stuff all my life, I would have had a different life, because I was an atheist for a while.
Wow. Wow. It just occurred to me, I have a few more slides, even though it's not going to be my.
Okay.
I'll say
Yeah.
the stuff on ALUs and adenosine, inosine.
Oh yeah. Please continue on the ERVs for sure.
But the signs, I don't have really good slides for what I'm about to show you. Oh, see, it says right here. This is the one. It says CTCF. So the ERVs, some of them have CTCF. I wouldn't say, since it's kind of like, this is kind of an afterthought. So I wouldn't say there are a lot of them that have it. But there was at least a little bit. The more important one is the signs. And I don't have really good, I have to rework these graphics because you're going to love, I think you guys are going to love this. This was actually discovered by Francis Collins. Francis Collins, and the evolutionary biologist that the NIH fired. Do you all know about Richard Sternberg? Anyone want to talk about Richard Sternberg and introduce it to the crowd?
I have brief knowledge of the subject, but I don't want to.
Okay.
I know I don't know enough to really describe it.
Richard Sternberg, 2004. He had a degree, a PhD in evolutionary biology and a PhD in another discipline. He has two PhDs. He's working for the NIH and simultaneously doing work at the Smithsonian Museum of Natural History, I think, or one of the Smithsonian's. And he was editor. He let a paper by Stephen Meyer get published. He was the editor, and they savaged him. They destroyed his career because he let.
Horrible.
Yeah.
They literally escorted him from the building, didn't they?
Oh,
From the
that's
office.
bad.
He did a lot of
Are
nasty.
you joking?
No, I'm serious.
What? I thought it was a joke.
At least in the version I heard, Stephen Myers talked
That's
about
terrible.
it. I remember him saying that he literally got shown, the whole in the movies, the guy hands you your box when you're fired kind of thing. It was that kind of analogy of what happened to him in terms of being the publisher or the editor. Sorry.
Yeah, it was mean. He's an evolutionary biologist, just because he let Stephen Myers suggest intelligent design. That was intolerable. And so okay, maybe don't make that mistake again, but you don't kill someone's career over it. It's maybe a little reprimand, but not take his life work away. I mean, I don't think that, given all the stupid ideas that we get from evolutionary biologists, that seemed like a very minor thing, because evolutionary biologists are pumping out things that are eventually shown to be false. No one gets fired. So anyway, he got punished for that. It resulted in a congressional investigation, but it fell outside of their jurisdiction. But they had special counsel to everything, and they weren't able to help him. So this was a big deal. So he still kept plugging along, and thankfully he continued research. He continued research because he's a true scientist. Didn't matter how bad his circumstances were. He wanted to learn more. He was seeking after the truth and discovery. So this paper by Francis Collins found a very interesting pattern in mice and rats. So let us hypothetically, and I'm not a baraminologist at all, okay? I don't go there. But let's just say hypothetically mice and rats shared a common ancestor. So we're just going to use that as an assumption. So Sternberg and Collins are assuming that mice and rats shared a common ancestor. So just imagine we have this book called Jane Eyre, or something Ancestor Eyre. Just call it Ancestor Eyre is the book. And then we end up with two books, one book that has Jane Eyre and the other Mary Eyre. So everywhere in the book called Jane Eyre, where there's the name Jane, the corresponding book will have that name changed to Mary in every passage in the book, all hundreds of pages or whatever. So we would reasonably suspect that the two books may have shared a common ancestor if not one evolving from the other. Does that seem reasonable?
Sense.
I mean, if we were just given this book and then this book, we could say, hey, we think that there was it descended from the same idea, not physical common descent, but just descended from the same idea, at least in terms as far as books go. So maybe I have to work on the analogy a little better. But what happened was let's just say we had mouse. Yeah, we had mice and we had rats. So we'll just assume that they physically descended. But what happened was the signs in the mice were spelled one way and the signs in the rats were spelled another way. And what was really bad in this paper that Francis Collins raised up was that they said, well, these patterns would have to assume that the mouse and rat descended from the same common ancestor, and then afterward, the signs in the rats and then the signs in the mice came afterward, because the signs in the mice are spelled differently than the signs in the rats. So far, not so bad. The really bad part, before I get ahead of myself, the really bad part is they're positioned into the same parts of the genome. That's what Sternberg found. And the ID community that could understand what he wrote, it started to send shivers. They said, this is an awesome
that means that they all had to be related. Created, for them to be in the same place, right?
Exactly.
Yeah.
This just blew people away, and it got buried away in some obscure thing. And where this relates is, guess what? Many of these have CTCF motifs that make the chromatin loops.
Oh, wow.
So I wanted to end on that. And, before I go on, I'm just going to end on that if people had
great.
questions. So this was the crowning moment of the transposon discussion.
Well, it was very interesting. Before you you went on to the piece that talked to you of your little the text there about the global search and replace. Before you popped that up, that was what was going through my mind. Of, did a find replace function in the code? That's literally, I kid you not, that was literally what was going through my mind. Of
right
when you were describing that. Well, that would make total sense if you're just doing that. So the spelling is different. And I'm assuming it results in modified regulation. Is that we're kind of in the variations?
I think I don't know. So first off, I don't know. But the suspicion is this is a convergence. They have the same function, but it's spelled differently. Now, I haven't had a chance to look at the CTCF motifs if they're identical.
So from an evolutionary perspective, or the evolution perspective, it would require, you're saying there it's in 300,000 different locations. Per the evolutionary model, somehow in 300,000 different places, the exact same mutation had to have occurred.
The exact same insertion mutation in the general same location. I mean, it's not 100% identical, but this is what they did. They
minor variants, but basically in the exact same spot for all intents and purposes.
Right.
These hyper-specific point mutations, if you will, would have had to have occurred.
Right. This is what this diagram is pointing out. And so if you Google that mysterious sign signal in Sternberg, it's going to pop up. You'll see. And the reason I like this is Sternberg is not a creationist. I don't know if he is now, but he's a trained evolutionary biologist. This is his bread and butter. He understands this stuff. He knows, that's why it was just so beautiful to read it. I said, this is from an insider. Yeah.
So have there been any, is this one of those instances where the staunch evolutionists just don't even admit that this has been discovered, or have they tried to come up with some sort of creative explanation for how it's possible?
this is how they deal with it, and this is why I said watch for the red herrings. They'll say, "Oh, how do you define function?" So the conversation will be about stuff like that. It'll never be about stuff like this because they don't want to talk about stuff like this. And a lot of them actually don't understand it. This is I don't mean to be mean, but
be mean as you like.
I'm sorry.
Be mean as you like.
So be as mean as you would like
to.
Oh,
sorry, I didn't realize I did have audio problems. I'm sorry.
No, you're Australian.
I see. Yes. Yeah, I had that. I've been to America twice, once and LA, and I've been to Hawaii twice, and I've had people ask me, like I said, fly, which is fly in American, and I kept saying fly, and he kept going, what's a floor? He couldn't understand fly, so I had to spell it out, and then I thought to myself, I went, oh yeah, fly, and he went, oh, flies, like that. So yeah, I can understand you not understanding me. Yeah.
So a quick question for you. So on these regulatory loops that you've been showing to us, do you happen to know if they're discovering any kind of there may be a similarity in the DNA, but have they discovered any like in between organisms, right? Are they discovering any different variations of the actual regulatory loops that are being expressed?
I don't know. See, we're only beginning to. That's why the 40 nucleome is just like this is new territory, and we're barely able to even understand one organism, which is the human species. This is really tough. We're getting to the point this work is really, really tough. And there's not going to be any end of the work. But I was about to say I'm going to be really, this may sound really mean, but I talked to evolutionary biologists. I said, I think these are PhD evolutionary biologists. And I'm just like, you don't know that? You mean you've not heard of 40 nucleome? Or, you don't know. I'll tell you what's really embarrassing. I one time said, chromatin has readers, writers, and erasers. And this evolutionary biologist said, you're just making that up.
Well, so it's actually interesting you say that. A couple months ago, I actually had an interaction with a PhD from Vanderbilt. When you're mentioning your friend, or the general, not your friend, but your co-researcher. It was interesting. I'd love to have a conversation with him because I forget the guy's name that I interacted with, but PhD in human genetics at Vanderbilt, and he and I had a conversation, and there was a plethora of things that I was bringing up to him that he didn't know, and my mind was rather blown because I don't consider myself to be any level of expert, and different things I was putting forth to him. I'm like, you're a PhD in genetics and you don't know this? That was kind of what was going through my mind of how is this possible? And what was really at the time, what was coursing through my mind of was, have you been put? Do you have blinders on in the context of what you would even consider to be reasonable trains of thought in your research? It was kind of what was going through my mind.
Sal, just quickly, Sal, are you tired because you said that you'd end there and you had a sore throat before? I mean, if you like, we can end this and we can go on another time. But if you're happy to go on, then that's fine by me. But I think that, because you had that sore throat and everything, it's what do you want to do, Sal? Do you want to end it?
No, I wanted to take any audience questions. They've been so patient and silent.
Can I get a question in?
Yeah.
Yeah.
Hey Jay, what's up, John?
Hey,
thanks for letting me in, guys. I appreciate it.
No problem.
Yeah, just to go a little off topic, but I know I'm probably beating a dead horse here. You had mentioned the chirality problem to snake. I know this is a big problem in the atheist community. They don't want to discuss it. If you could give me some points on that, the major protein problem that they're having with selecting all left-handed amino acids. There's a lot of denial, and I figured, you're probably pretty well versed in that area.
Oh, thank you for asking. And who am I Who am I speaking with?
I'm Tony.
Hey, Tony.
Yeah, real pleasure to talk with you, man. Really
Thanks.
appreciate it.
There is some, the evolutionists will say they have literature that proves that, it can be solved. That's not completely honest, because life solves that problem. So there's definitely there's kind of chemistry that can solve it. There's not question about that. It's whether it can happen in a real context. So the homochirality for amino acids, yeah, we do have some reactions where we have reactions where we can get all 100% left-handed or all 100% right-handed when exposed to a catalyst or whatever.
Right.
And they'll have these papers and say, look at this. This proves that it'll happen. And
well, it's an intelligence
Would
that
you-
added the catalyst and then stopped it at the, that's
Right.
funny.
Would you say that that's a controlled laboratory environment, or does that relate to the early Earth in any way?
well, that's the first line of argument. But then I found one paper that called them out on it. It wasn't written by a creationist. It was written
Great.
by origin of life researchers, which is always the best.
We love evolutionist
Yeah.
citations. We
Yeah.
love them.
Love it.
And my other lecture on Friday, my other talk, actually cited it. I can't pronounce the Russian's name, Konstantinova or something. And anyway, but they said, the problem is you have one catalyst that's going to make them all left and another catalyst somewhere else that makes it all right. So
Right.
what happens is if you have all these catalysts mixed together in non-controlled conditions, what do you get?
A mess.
Yeah, you'll get a racemic, which racemic means, mix of left and right. So that's the first problem. This is just amino acids. It gets really bad with, I wouldn't say it's a knockout proof of creation, but it's making it tough.
Would you say, I'm sorry, but would you say, because they always say that we're the ones that are in denial and being very dishonest when it comes to the chemistry, but would you say that in order to prove abiogenesis, you would have to start with a random pot of chemicals? I would say the atheist is being totally dishonest, and these origin of life chemists that are selecting the exact molecules they want to be in their experiment. So of course it's going to be successful.
I think they're being dishonest with themselves. I think some of them are sincere. They sincerely believe. I mean, a person can be deluded and not believe what he's saying is a lie. There are people that know what they're saying is factually untrue, and they'll do it anyway. That's definitely a lie. If someone's just mistaken and deluded, I try to cut them space. But can you state your question again? And help me with your name. I'm terrible with names.
Just Tony.
Tony.
Yeah.
So what was your question again?
The severity, how serious of a problem is the chirality problem? And in other words, the atheist is always calling us dishonest when it comes to the chemistry. As far as I'm accusing them of starting with the chemicals that they want. In other words,
yeah.
They're always selecting what they want to be in their experiment, intelligently guided.
Okay, if I were defending the argument, I would say it's serious, but not quite yet a deal breaker. Although it's really close to being just like enough.
Yeah,
it's close enough. But there's one other thing that they don't like to mention is even supposing you had all left-handed at some point, and some of the ways that this can be done is through drying. So the amino acid, one of the experiments is they had boiling water, they threw the amino acids in, and then they let the solution cool, and then they let it dry, and it would start to crystallize. And then it would crystallize either all right or all left.
right.
I said that was an interesting experiment. Of course, they don't tell you that, what happens if the ocean washes it and mixes the two back together? That's too bad. You lose all the homochirality. But there's one other thing. Let's say it's enough that you get just a nice little pool of these left-handed amino acids. Well, how long is it going to wait around to connect? First off, if it's in water, that's really bad because it has a hard time connecting to make these proteins or even a protoprotein. but as it waits around, the left will spontaneously flip to right.
Right. They switch.
It's what they call racemization, and
Yeah.
it gets worse with temperature. So I don't know, there's been some debate how long this can last.
Like
By
when
the way,
meat gets burnt. Like
this is,
racemization, when meat gets burnt by a flame, then it binds up the proteins, and that's what's racemized. That's what's bad for you. It's toxic too, isn't it?
yes. And by the way, just as an aside, this is an interesting dating mechanism. This is a project I'm actively, passively working on when I can squeeze in the time. The
Great.
racemization state of the fossils at the lower levels, the lower strata, is about the same as the higher. This makes it look. So in addition to carbon-14 and collagen dating, we have racemization dating. And this was not my work. Someone had done this in the mid 80s, and no creationist had taken it up. And I've said, we ought to pursue this now. If we could just get a chemist to work on this. This year I've been in the process of gathering the data for the Institute of Creation Research, because I want someone to take off on it.
Sal, would you agree with somebody like James Tour, by the way?
Oh yeah, on a lot of things, yes.
Okay.
Hey,
yeah, you were pretty direct with Snake. I wish you guys would be, I don't know, a little more, kind of get to the point with them, because the delusional internet atheist just gets away with too much nowadays, I think.
Yeah. Well, and it's good to see Snake humbled the way he was. While he was speaking to you, he wasn't so cocky and cheeky.
Yeah. You have the intelligence to put these people in their place, and that's what they need.
Yeah.
Well, the thing is, I'm just going to do my job and say, if we get him shouting match, that'll it's not a good service to the listeners that
No.
They want to keep hearing. I want to be able to, so a lot of times I'll just say, let's, we're going to disagree on this. Can we just talk about something else? Because we're not going to resolve this. So can I talk about another fact? So I was just climbing up the stairway of life. So the homochirality problem for amino acids is really, really bad. I wouldn't call it a deal breaker. The homochirality for nucleotides is really bad. It's homochirality and being able to form them. I mean, it's, with amino acids, it's maybe left or right. With the nucleotides, it's like combinations, when you're hooking them together, it's like 16,000 different combinations for just two nucleotides. 16,199 ways to get it wrong. One way to get it right. That's just the first two. I said, just try doing that for a polymer of 10. So let's say you're trying to get 10. Okay, you could see the explanation. Yeah, it's just nasty.
And what it takes to make to build a working protein is, and just a working anything is amazing. I mean, I've watched Stephen Meyer, give the math from Douglas Axe on things in detail and that, and it's absolutely mind blowing.
Do you know about the RNA on clay? There's a lot of atheists that'll bring that up, but James Tour was saying that the clay is chemically prepared in a way that would never be found in nature.
Well, there's the other thing, that homolinkage thing that I described. Those RNAs have to be the right chirality, the nucleotides and the phosphate group has to be put in the right position. There's 16. If they have an adenosine monophosphate linked to a cytosine monophosphate somewhere in that RNA, which almost will be inevitable, there's 16,199 ways to get it wrong. They had to pre-prepare those RNAs. And it's really hard to get that right. And in addition to what James Tour did in my talk, I've referenced the work of a physical organic chemist. That's her specialty, Change Tan. And she went through this and said, well, this is exactly why it's so hard to build this in the lab. We have to use living organisms to help us build this, because we can't just make our own chemicals and start building it from scratch. We don't have the technology. When they're trying to build their own DNA, where you didn't have a template like you do in biology, if you just had, like, say, you wanted to, like Craig Ventnor was doing, was trying to get it from a computer, and then we just build it from scratch in the laboratory with beakers or whatever, they could only get 65 or so DNA bases together. They couldn't get like the 500,000 that you need even for a minimal genome. 65 they could stitch together, and they had to use all sorts of chemicals where you'd get the reaction, you'd put the chemicals there, you'd stop the process, you'd change the temperature, put a different set of chemicals, and you build
Wow.
one nucleotide. And then you
Amazing.
go to the next nucleotide.
Wow.
You
How
repeat the
does that
process.
happen in nature?
Right.
It's not.
That's
Forget it.
amazing, man. That's incredible.
And that's so you, and what's really bad is that assumes you have the right nucleotide to start with, or you have the right phosphate group with the right
So who selects those molecules just to get that experiment going in the first place? The intelligent chemist. Am I
Yeah.
wrong?
Yes, and they're not that smart relative to the one who made it for original biology. Because
Yeah, God.
when they did it for biology,
That's right.
it's almost laughable because it goes these things. It's really funny. They could get it to 65, and they didn't have very good proofreading. After that, they had to sort of give up. But what was really, there's some other problems. If you just let DNA just sit there in water, it has all sorts of nasty reactions, depurination, and then just spontaneous breakage, and that's why you have DNA repair mechanisms in your body, because in your body there are 3,000 depurinations every day, and
Yeah. We've gone in no
your
time.
body would be blowing apart. And
Yeah.
I'd be curious to see you debate somebody like Aaron Ra. I don't know if you're familiar with him. He's pretty much the internet atheist leader.
Oh, everybody knows
I had an
him.
exchange with him once. It was polite,
Wow.
and he was,
Polite.
I'll tell,
Really?
can I share how that one
That's
went?
amazing.
I'll tell you
We
how
want to
that.
hear about this. Sorry.
I'm sorry.
I'm sorry I cut you off. Sorry.
Oh, I'll share how that one went. It was a cordial 10-minute exchange, and I was identifying myself to him, and I was saying, I work for John Sanford, who, he invented the gene gun. He's very famous. He's an applied geneticist, and he's a creationist. And Aaron Ross said to me, "Oh well, maybe I could teach him about evolution." I just like, okay, this is not going to work.
Yeah, denial.
He said,
He's going to educate everybody
Why
because he's the
don't
authority.
I send him? Why don't I send
He's-
you? Yeah.
He's a used
Aaron-
car salesman.
Huh?
Yeah.
He's a used car salesman. He sells used cars.
More like a clown.
So he said, "Why don't I send you a PDF file, and you could share it with Dr. Sanford, and he'll understand why evolution works." And I was trying to explain how I said he used to be an evolutionist. He's a geneticist. A big fraction of the gene. He just ignored everything I said.
Yeah.
Yeah, you're gonna
It's amazing.
teach a Cornell professor that he doesn't understand this stuff.
Yeah, who invented the gene gun for
Yeah.
plants? And I mean, it's like, yeah, I know. It's so arrogant, isn't it? We bump into that sort of thing all the time, and we post stuff, in detail and explain it, and we get the we in our papers, in our books, we use mostly evolutionist links, and we explain it in great detail, and they just you accuse you of all sorts of things, but yeah, it's amazing how they just turn a blind eye to just about anything that they won't even look at it.
Well, I want to cut some slack. There's some, there are people that are, maybe the Lord will turn them around. But then there are
You're
people,
right.
yeah, there are people that are diehards, just like the Pharisees. They could
Right.
see, they could see
I've
Lazarus
persecuted
raised from the
Christians.
dead, and they'll try to kill him. A lot of logic in that, and.
When I was an atheist for that time, I persecuted Christians, not badly, not beating them up or anything, but just calling them names and stuff like that. So I understand where evolutionists are coming from, and atheists, and I know how they think, because I lost my faith when I was 10 over the vestigial tailbone, with images of swinging through a tree, and with my tail, and yeah, and then it was like till a lot of time later. So. Yeah, I understand what you mean.
Hey, Sal, quick question for you.
Yeah.
Some of it, the stuff you were talking about earlier on the addresses. I remember a while back, I forget where I read this, but they were talking about proteins that are exported to other cells. Also have addresses. built into them. Is that correct in terms of like where they end up in a completely separate cell or a different part of the different system?
Yes, and I have a 2-minute video that will. I mean, if you're interested, it's only 2 minutes because it can explain it.
Yeah. If you want to, yes.
Let me share it. Oh my goodness, this is
Yes.
why a prokaryote cannot evolve into eukaryote. So let me get it. Let me see. I'm going to share my screen. And Praise, thank you for helping me out here. And hanging.
Yes, thank you, Praise. He does a wonderful job. I'm going to get myself a nice computer soon, and he's going to teach me how to use it, so I can give him a break. He really helps out a lot. Praise I am that I am. Yeah. Just take your time, Dr. Cordova, as long as you need to get the thing up, the video up, sorry.
Can the screen be shared?
Hey, Praise, is it
Hang
you
on.
authorize sharing? He may have stepped away from his computer.
Uh-oh. He might have gone to, I don't know what time it is where he is. Praise you. Oh, no. There. Oh, he must have thought we were. He must have thought we were.
He must have stepped away.
Yeah, he must have stepped away. What a shame. Oh.
So can you give us the brief
Well.
the brief run? Next time we'll play the whole video. Can you give us the brief rundown on that? Because I remember when I came across that and was learning about that concept. It really kind of blew my mind. And what you've been talking about tonight on the addresses of the zinc fingers and such. I'd love to hear a little bit more about how that works. I think you're on mute.
Sorry. Thank you. We call them localization signals, and we have import and export. And so, first off, I want to thank Snake and all the people that have talked to me that are on the other side, because I don't want to, or even Jackson Wheat, I don't want to trash them, but I will point out when they had a bad argument. And Jackson Wheat, one time I said, how did membrane-bound organelles emerge in eukaryotes? Because if you just take the nucleus, how do you get stuff to go in and out? And he said, leak in and out. And just like, okay, I didn't call him on it, but I just like, this is, I'm just going to let that one go.
Yeah, that's hilarious.
I'm let that one
just
go.
leaking it out. Oh man.
We're not going to, I mean, if you can believe that, I'm just like, okay, we're not going to, we're just going to have to talk about some other things. And so no hate intended, Jackson.
No. I like Jackson Wheat. He actually came onto my channel once and one of the evolutionists did terribly in the debate, and he actually admitted it. So good kudos to him for doing that one. Good on him.
Yeah.
So, we appreciate it. Anyway, sorry.
Yes. So the reason I can't do that is that it the molecules, the proteins are actually too big to fit through with diffusion, what they call it's just going to leak out. It has to be actively pushed, and the video actually went into all the proteins that are involved, and there's several of them, and it takes energy. It takes this GTP, and then it has these proteins called importines and exportines, and it looks for that localization signal. So all these proteins have to be formatted that go in and out. They have to have that motif in them. So this is how you construct. How do all those? If you're going from prokaryote to eukaryote, and now you have a nucleus, you have all these proteins that have to go in and out. So all these proteins that were okay in the prokaryote, they all have to be simultaneously formatted with these localization signals so that the protein goes to the right compartment. That would entail having numerous point mutations simultaneous, or some very fortuitous insertions of a nuclear localization in all these DNA regions. for no good reason. At the same time, when importeins and exporteins and these transmembrane proteins emerge, I'm just like, I'd sooner believe in a miracle.
Well, yeah, when you're talking about the signals there, I remember recently I was reading some stuff on retrotransposons, and I think that's the GAG protein they have to have in order to go through the portals. I think there was several other ones, but I remember reading something. It was like the GAG protein has to be associated with it, otherwise it can't go through a portal. And thought it was very interesting in the concept, especially with all the the Dawkinses of the world and their jumping gene arguments. And I'm like, yeah, there's like all these additional external variables that are required for that to what you act as if is just this easy to occur modification. There's actually a plethora of additional functions that are required to make what you're taking for granted happen. It really is interesting to, the more we learn, the more things you're talking about and that are being discovered, how anybody can argue this is all just random occurrences. When you were just mentioning who knows how many simultaneous point mutations would have been required. But your argument from the evolutionary perspective is that it's a slow, steady process, gradual process. But it requires simultaneous execution in order to change. It's like the direct antithesis of each other, yet it's being argued that, yes, this happened.
If you saw me looking to the side here, I was trying to find out the names of those proteins. They have one called RAN-GAP. I don't know what the one with the GAG protein does. So that's why we need Matt. He can set us straight.
Yeah.
Hey, Sal, I just wanted to ask you, what would you say are the top 3 death blows to abiogenesis? What would you consider the most serious problem with that, hypothesis?
Oh my goodness. Top 3 is hard because there are at least 12 that are really, really hard, and they're almost all equal.
Okay.
And actually, homochirality is probably one of the ones that aren't the most severe. So I would say the homolinkage one with the nucleotides is really bad. It'll blow away the RNA world. It'll blow away any. That's
when you bring that up to an atheist, I mean, do they ridicule you? Do they say you don't know the chemistry? You don't know what you're talking about?
Oh yeah, you can imagine everything that they could say. One professor used to bully me a lot, and he still does. And he really hates it when I call him out on what he doesn't know. And he's a professor. So those are the best. Yeah, especially when I show him up on basic chemistry, because I know it's probably been a long time since he took it.
So what's their answer?
So he'll say it. Oh, I'll tell him by name because he said some nasty things. It's Arthur Hunt. He debated Stephen Meyer, but Meyer doesn't bother with him anymore, so he's relegated to debating me on the internet. He had the gall to say, you should go back to apologetics.
I feel sorry for him. No, I don't. I was joking. I said I feel sorry for him. No, I don't.
Oh.
I know my accent bamboozles you. I'm going to actually talk a little bit, space my words out a little bit, because when I string them together with my Australian accent, it must be really hard for you to understand.
So earlier when you were talking about the minimum 500,000 nucleotides for life forms, it's interesting you brought that up. I did the math one time. I think it was obviously the base of that is 4 to the 500th power. I think that if I remember correctly, I think that translates into 10 to the 52nd. And they tried to argue, the person I brought that up to tried to argue that you can't. That's not reasonable.
I need to take a commercial break. I'm going to shut off my cam for just. I'll be right there.
Yeah,